Hi everyone, a very good evening to all of you. I really hope my audio and video, everything is okay. Hope you guys are holding on well. Right now, obviously, you all must be tensed. The exam being near, plus on top of that, the current situation that is happening with the number of years you have to do your internship for. Definitely it puts a stress on your head, but it's okay. These things will keep happening irrespective. We will have to swim through. So, ठीक है. Now, before this session begins, let us take few words of motivation and then we will go ahead. All right. This is basically one sher shayari I want to say before I start, and this is on your current situation. All right, so let's start. साहिल का सुकून से किसे इनकार है लेकिन साहिल के सुकून से किसे इनकार है लेकिन तूफानों से लड़ने का मज़ा ही कुछ और है. So this is pretty much it. Exams are that situation which gives you that thrill. Obviously, we don't like that, but the adrenaline rush that we get inside while we are entering the hall, okay, hopefully you go in with that kind of enthusiasm and comes out with success. Okay, all you need to do is have a decent amount of confidence. Any exam of the world is going to be pretty much similar. Okay, so do not make this too much of a fuss. हो जाएगा, ठीक है. कर हौसला बुलंद, तू ना घबरा, कामयाबी मिल ही जाएगी, तू छलांग तो लगा. Okay, so this is how it works. All you need is some kind of motivation inside you to keep moving forward, and baki sab the story will fall into place. Okay, so anyway, welcome everyone. Now we are going to begin our session, start from now. And please know this is a rapid revision. I will not be going into the intricate details of the subject. See, for that we have our regular classes. Abhi bas ye last minute revision mein what are the important points, that's what we are going to cover up. That's it. ठीक है? Now, starting with the first things in the general microbiology, a very important topic is toxins. So now to understand the toxins, what we will do is we'll just draw a box, and this box will represent bacteria. Now you need to understand there are two types of bacteria divided on something called gram stain, which are gram positive and gram negative. And here you are, there are two types of bacteria, one that is positive, one that is negative. Imagine this guy is gram negative, this guy is gram positive. Okay. Now we are talking about the toxins that are released by the bacteria. Now, one type of a toxin is such that it is released outside the bacteria. Since it is released outside the bacteria, we call it as exotoxin. And then there is another one. This is where we have, which is inside the bacteria, and this is going to be known as endotoxin. Simple logic. Endo means inside, exo means outside. Now, if you watch this picture closely, the way I have drawn this, you must analyze one thing. See, in this picture, you can see that both the bacteria release exotoxin. Exotoxin is the one which is excreted outside. And then only one bacteria called gram negative, it is having endotoxin. So endotoxin is an exclusive feature of only gram negative bacteria. Okay. Whereas exotoxin is secreted by both gram positive also, gram negative also. Next thing, exotoxin, it is made up of proteins, whereas endotoxin is made up of certain sugars, what we call as lipopolysaccharides. Okay. Next, moving on, one guy is heat sensitive. You can also call it as heat labile if you want to. And another one is heat stable, also called as heat resistant. Now, what is the reason we are learning all this? Like, toxin के बारे में जानना ही क्यों है? Why should I know about this? See, simple reason is this. Imagine if the disease or the infection, it is produced by the toxin, then you might have to produce a vaccine against it. And for the vaccine, you need some substance. भाई आप कुछ तो डालोगे ना उस vial में. You have to put something into that. And you're going to put some toxins which are causing the diseases. That's what we're going to put inside. So now if you check this out here, exotoxin is more easily collectible. We can collect this exotoxin because it's excreted outside the bacteria. And what can I do? I can heat it. And you know, whenever you heat something, it becomes much weaker. Once the toxin becomes weak, you can inject it into some human being. And that guy, once this toxin is, injected into that guy, he will produce antibodies. In other words, he's going to produce immunity. So, sir, this exotoxins are those ones which we can collect, heat it up, that is why heat sensitive, then we can make a vaccine out of it. The vaccines which contain toxin inside them, they are called as toxoids. Example, TT. TT ka full form itself is tetanus toxoid. Tetanus toxoid. Yes or no? And another one, DPT. DPT is also into this category where you are having toxins of these bacteria. See, these are the diseases which are not directly caused by the bacteria, but rather by their toxins. DPT, diphtheria, is not directly by Corynebacterium diphtheriae. It is by the toxin it releases. Pertussis, it's by the toxin. Tetanus, it's by the toxin. So any disease which is by the toxin, we need to produce vaccines, and for the vaccine we need toxin. और वो कौन सा toxin है which we can use for production? Exotoxin. And thankfully exotoxin is produced by all bacteria. That's why we can produce the vaccines against all bacteria. So that's it. Now that we are clear, let's move on. Mechanism of action of toxins. Doctors, what do we do to kill bacteria or organisms? We create antibiotics, right? We create antibiotics. तो फिर bacteria भी कुछ करेंगे. They are not going to sit back. If there is an injustice done from one place, other people will retaliate, just like this. If you are killing them by making antibiotics, the bacteria are making toxins to kill you. That means we are learning the mechanisms of toxins which can damage human beings. All right, very important topic. The first one is protein synthesis inhibitors. That means these are the bacterial toxins that come inside the human body, and what do? They stop our protein synthesis, which may cause some problems. Okay, first one, diphtheria and Pseudomonas. These are the two guys which work on something called elongation factor 2, EF2, elongation factor 2. So elongation factor, this is something that is required for making proteins, to elongate the chains of proteins. So that is what is blocked by this guy. Okay, so हम इसको ऐसे याद रखेंगे. If you know the full name of diphtheria, diphtheria is called Corynebacterium diphtheriae. Yes or no? Corynebacterium diphtheriae. So you may remember CDEF. ठीक है, या तो आप ऐसे याद कर लो. One way I'm giving you CDEF, like that you can remember. Or another way that I'm giving you here is this. You can remember di and the word do. Di means two, do in Hindi means two. That means EF2 is blocked by two guys. One is diphtheria, another is Pseudomonas. I hope everybody is clear with this. Okay? Now, next thing is another way bacteria damages our protein synthesis. They damage our ribosomes. Okay, they damage our ribosomes. You all know humans have two types of ribosome. One is called 40S, another is called 60S. ये हमारे ribosomes है. These are our ribosomes. And what is the bacterial toxins doing once they enter inside? They are damaging our ribosomes. So there are two guys who can do this. One is called Shigella. You can remember S for 60, S for the Shigella. And another guy is called EHEC, Enterohemorrhagic E. coli. Enterohemorrhagic E. coli. Please remember, this is going to be also known as Shiga-like toxin because it's ditto. The presentation is going to be bloody diarrhea by both of them. These are both the guys that produce bloody diarrhea inside us. Shigella dysentery. Dysentery is a bloody diarrhea. Another is EHEC, Enterohemorrhagic E. coli. सो टेक्निकली आप लोग इसको S से याद करोगे, ठीक है सर. नाउ देयर शुड नॉट बी एनी कंप्लेंट्स, ये कैसे याद रखूं, क्यों याद रखूं, एग्जाम पास करना है. नाउ यू नीड टू पास द एग्जाम. वी आर जस्ट लर्निंग वेज हाउ टू रिमेंबर इट. आफ्टर एग्जाम you forget it, nobody cares, right? So I am just telling you right now how to remember. S for 60, Shigella and Shiga like. Shiga toxin and Shiga like toxin, S for 60S. Now next, moving on, you have some guys called neurotoxins. Neurotoxins. Now what are these neurotoxins going to do? One is called Clostridium tetani, one is called Clostridium botulinum. Each of these guys work exactly opposite to each other. They are going to work exactly opposite to each other. What do they do? Sir, all of us know tetanus itself means contraction. Yes or no? So Clostridium tetani will produce contractions and Clostridium botulinum will produce relaxation. Exactly opposite. So how exactly are they working? Check this out. Sir, we all have one neurotransmitter called GABA. GABA is an inhibitory neurotransmitter, so it relaxes your muscles. I repeat, GABA's main function is to relax your muscles. So if you block GABA or another neurotransmitter called glycine, now your muscles will contract. Does that logic make sense to you? GABA ka function is to relax. So if you block GABA, now there's going to be contractions. Same way, how does this botulinum dude produces this relaxations? Okay, now let's look at botulinum. Sir, botulinum works on this thing called as acetylcholine. Okay, botulinum works on acetylcholine. What is the job of acetylcholine? Let's just check this out. Acetylcholine contracts muscles. So if you block acetylcholine, there will be relaxation of muscles. Okay, there will be relaxation. So you must remember, botulinum will relax the muscles and tetanus will contract the muscles. Done. Next is cyclic AMP inducers. Now you must first keep in mind, any bacterial toxin that increases cyclic AMP, this is either going to manifest in the form of edema or in the form of diarrhea. Now even when I say diarrhea, which type of diarrhea? That has to be very precise. You must say watery diarrhea. Watery diarrhea. Is everybody clear here? Cyclic AMP को बढ़ाने वाले, somebody who increases cyclic AMP, they will either cause edema or watery diarrhea. So guys, now understand this. First of all, how to remember which toxins work here? The word cyclic AMP itself is teaching you that. C for cholera, C for cholera, A for anthracis. Okay, A for anthracis. Next, P for pertussis. The only one that we are left out with, you need to play a little, is this M. M को थोड़ा सा घुमा लो तो E दिखेगा ना दोस्त. M, if you are turning around, it looks like E. So we are going to kind of memorize like this. E-tech, E-tech, E looks like M. Okay, so cyclic AMP, this is the story. 4 toxins that can increase cyclic AMP, either they will cause edema or they will cause watery diarrhea. Sirji, I am going to use the purple color for edema agents in this. Anthracis and pertussis, they will cause edema. They're going to cause edema. And then now the next part says cholera and the E-tech, they are going to cause watery diarrhea. Okay, they're going to cause watery diarrhea. Now, one more additional point that you must keep in mind. See, anywhere you must have studied, you must have learned cholera produces rice watery stools. Rice watery stools, yes or no? So, वो तो अब समझ आ गया आपको कि watery diarrhea होगा. Now, I'm adding one more point that they may ask you. In our GIT, precisely in the luminal area or the lumen, there are certain receptors. These receptors are called GM1 ganglioside receptors. Okay, GM1 ganglioside receptors. So where is cholera going to bind and produce its effect? It will do it from GM1 ganglioside. When it binds to this receptors, there is obviously a whole lot of detail in this that finally increases cyclic AMP. Cyclic AMP. and once the cyclic AMP increases, it will be producing watery diarrhea. So this is the logic CAMP. यहाँ पर the words we have learned. Okay, cool. Now, next part, next section here, you're looking at a topic called bacterial growth curve. Now, when you look at this, what exactly you must first understand is, where are we talking about the bacterial growth? Are we talking about this in the human beings? The answer is no. We are talking about this in the culture media. Okay. For humans to survive, we need food. Same way, bacteria also to survive, they also need food. And we are just providing them with food in the culture media. Yes or no? So, the amount of food that is given in a culture media is very limited. So, that's why the bacteria cannot live forever. They cannot multiply forever. What happens, as long as the food is present, they will be living. But after the food has exhausted, they will die. So that's why in the culture media, there will be one time where you will be seeing something called death phase. Okay. So here we go. First, once you have put an organism into the culture media, it does not start reproducing right away. It undergoes this first thing called as lag phase. Lag phase. All right. Now what happens is, once the bacteria realizes there is so much food available for its reproduction, now it cannot hold itself. Suddenly it will start doing the things that you like to do. Okay. It will start reproducing. And this is where you must know it reproduces so crazy that we can't even count it. Okay. Hence the name is called log phase. Log means logarithm. Log means logarithm. Logarithms are those mathematical things that we used to see that cannot be calculated directly by our mind. That's why we used to carry log papers in our exams also. All right. So technically log means they're going to reproduce like this. And if anybody asks you what is the process by which bacteria may reproduce, you must answer one word called binary fission. Binary fission. That means each time the bacteria reproduces, they become double their original numbers. So this is log phase. Now suddenly the bacterial numbers have gone up, right? They have become very high. Up what happens is, after this time, suddenly there is too many bacteria that has been born, right? So now what will happen is that there will be competition for the food. Some bacteria who are getting the food will survive. Some who are not getting the food will die. And this is where the births equals deaths. Okay, births equals deaths. And this is where it's a bad situation for the bacteria. Okay, इनके बुरे हालात शुरू हो चुके हैं. Now, whenever the conditions are bad for the bacteria, to protect themselves, they will form spores. They're going to form spores. So, spore formation takes place in the stationary phase. And finally, once the food has exhausted, they all will die. This is the simple logic right here. Lag phase, log phase, stationary phase, death phase. Questions can come in wide variety on this. They might ask you, arrange the following in the right sequence. Lag, log, stationary, death. This is the sequence. They may ask you in which of the following phases sporulation happens or spore formation. Answer is stationary phase. They may ask you which is the best place. Where is the best place where we can collect the sample from? Let's say you're growing this bacteria on the culture. Okay. So from where are you supposed to collect them? Please remember, this is the best for stain. If you want to collect a sample, you should collect it from this area, that is from the log phase itself. Maximum bacteria are found. That's why it's the best place. I hope everybody got this idea. Okay. Now that we are done, okay, let's move on to the next part. ये आपको दिमाग में रखना होगा because pretty much every year there has been at least one question on the motility aspects. ठीक है. So first one is tumbling motility. ठीक है. Tumbling motility. Tumbling motility is seen by Listeria. Listeria. Okay. Tumbling motility, it is seen by Listeria. Sir, if we translate tumbling in Hindi, हम क्या बोलते हैं उसको, लोटपोट करना, yes or no? Like when you are on your bed, nothing else to do, this is what we used to do as children, yes or no? बड़े होके अलग काम करते हैं वो अलग बात है. Now the listeria, L for listeria, tumbling motility, लोटपोट. So now this is the first thing. Okay, next is darting motility. Darting motility, you need to know that when you're talking about darting motility, it is shown by Vibrio cholera, and the same Vibrio cholera also shows shooting star motility. And by the way, darting motility is also seen by one more agent called Campylobacter. ठीक है, Campylobacter. Okay, so now that you're clear, sir, हम camping को जब जाते हैं, whenever we go for camping, we usually take arrows, or imagine you're throwing a dart like this. So Campylobacter also shows this darting motility. See, majority students already know Vibrio cholera से darting होती है. I'm just asking you to remember the extra one now, Campylobacter. When you go for camping, imagine throwing a dart like this. Next thing is moving further, corkscrew. Okay, corkscrew motility. Corkscrew motility, it is shown by the organisms called spirochetes. Corkscrew motility, it is shown by somebody called spirochetes. I hope this is clear enough. And Dr. Saab, spiral organisms will move spirally. Fancy word for spiral is called as, corkscrew motility. Last and the final, you must know falling leaf motility. This is shown by Giardia. Okay, a parasite by the name of Giardia. So ये अच्छे से याद रखना है आपको. Now moving further, this chapter we are quickly going to glide through this, sterilization and disinfection. See, you must remember when you talk about sterilizing something, there are two methods. One is physical methods and the second is chemical methods. Okay, so physical and the chemical methods. Now sir, in the physical methods, there are things like heat. Heating something will obviously kill it. Radiation and filtration. Filtration is less common, that's why I haven't put it in this list. So heat and radiation. Then we have chemical methods that we are going to see. Sir, when you talk about the heat, there are two types. If we can use dry heat versus moist heat. Okay? Dry heat and moist heat, these are the two different things. Now if you talk about dry heat, see, we can burn something. We can burn something. So sir, incineration is burning something, and we do this for all the biomedical waste. Anything which is coming from the humans, we are going to burn it, and the human things are put into the yellow bag. Okay? I hope you're able to remember this. You must have studied this in PSM also. Very important. इस पे question आते रहता है. Red bag, yellow bag. ठीक है? So now that we saw this, the most important part that we are going to learn when it comes to our micro is hot air oven. Okay, hot air oven. ये एक चीज पढ़ेंगे अपन अभी, hot air oven. And second thing that we are going to study is autoclave. Now look at this. Hot air oven में the temperature is very, very hot. It is 160 degrees. And that too, the things are supposed to be sterilized for 2 hours. ठीक है? So you must remember that if the temperature is so hot, we cannot sterilize everything into this. Especially, we cannot use it for two things. What are the two things we cannot sterilize? One is plastics and second is rubber objects. Generally, anything which is plastics or rubbers, we cannot use hot air oven because the temperature is too hot. So for them, plastics, rubbers, you have to use something else. Okay, you have to use something else. Now, autoclave, I usually call it as pressure cooker of micro. Why? Because the concept is exactly the same. See, pressure cooker mein the things can get quick, basically cooked faster. So same with the autoclave, only 15 minutes of sterilization is required. Okay. Why? Because one new component added here is pressure. So temperature has been lowered. Number one, that is the good thing. Next, the amount of time I'm sterilizing also less. And then finally we have added the pressure into this. So ये temperatures याद रखना होता है for the exam. How to remember? Hot air oven is hot 160 degrees for 2 hours. And autoclave is much lesser. You must remember it is the pressure cooker of our micro. Done. Next one, sir. Radiation में we call the radiation or ionic radiation as cold sterilization. Why do we use this word of cold sterilization? Is because without heating we are able to kill things. That's why it's called cold sterilization, because of the rays. Because of the rays, I'm able to kill the organisms. That's it. That's why it's called cold sterilization. Okay. Next, coming to the chemical methods. Alcohol, our everyday use thing. The sanitizers that we are using, the hand rubs that we are using, they're all containing alcohols like 70% ethyl alcohol. Aldehydes, very important. Formaldehyde is very, very important for us. Halogens mein, the things that we use at home, right? First of all, you can see betadine is iodine. ठीक है, पहली चीज याद रख लेना, betadine is nothing but iodine. What is betadine? Also called as povidone iodine बोलते हैं. We call it as povidone iodine. And with the chlorine we have chlorite. Okay, with the chlorine we have this guy called as chlorite. Now what I have done is I've just put one table here to remember who are the controls that we used in all these procedures, so that it's easier for you guys to revise. Okay, ये table देख लेना एक बार. Next, this is something that you need to keep in mind. As I told you before, hot air oven can be used for a lot of things that you can see here, but we cannot use it for plastics or rubbers. Okay, we are not going to use it for the plastics or the rubbers. Okay, that's the basic thing that you need to keep in mind. Now, formaldehyde and hypochlorite, in my opinion, very important what they are used for. Okay, sir. Formaldehyde is a very high level of disinfectant. It can pretty much kill everything, including spores. Not just the organisms, it also kills the spores of the organisms. All right. So now what I'm saying is formaldehyde is first of all used for fumigation of OT. Okay. Formaldehyde is for, imagine, cleaning the operation theaters, sterilizing the operation theaters. And because it's a highest level of disinfectant, if you add it to a dead person's body, that body will not be destroyed. That body will not get decomposed. So if you want to preserve a body for a long time, we always put formaldehyde into this. So all the cadavers that you might have dissected on, or the anatomical samples, etc., they are all preserved in formaldehyde. Okay. So that's it. You must know formaldehyde. Next is sodium hypochlorite. Sodium hypochlorite. Sir, please remember, this is the MCQ they love to ask. This guy is used in disinfection of infectious hospital spills. So let's say something dangerous might have fell into the hospital. So that time I am going to use this sodium hypochlorite for cleaning it. See, usually if the spill is less than 10 ml, that means whatever I was carrying, let's say that guy fell, that is less than 10 ml, I just need to use 1% of sodium hypochlorite. How is 1% also written? 1 is to 100. 1 is to 100. That is what is meant by 1%. Okay. Now, this is the thing that they're supposed to ask you. Now, next is there is another one. 1 is to 10. 1 is to 10. Obviously, this is more concentrated. This is more concentrated. Are you understanding this? See, the thing that is concentrated will obviously be more strong, right? So, please be very careful what the question is asking. If they just ask like infectious hospital spill, let's say they mention mLs, less than 10 ml, we will be ready to answer 1% sodium hypochlorite. But what the examiners are doing these days is they're just confusing the students. They're playing with you. Last line mein subtle way they will write one word, which is the best way. Instead of asking the regular one, they are asking which is the best one. Sir, best to ye hoga na, 1:10 is more stronger. 1:10 is more stronger. Please remember. Okay, if they just ask normal, answer 1%. But if they are going to ask you best one, then you answer 1:10. Okay, so that's it. Don't fall for this trap. I've clarified it right before your exams. Okay. Next, sir. Moving further. Check this out. Now, here we are. You're looking at this thing, that is antibodies and their properties. Basically, that's all I'm going into now. Antibodies and their properties. As you know, there are 5 antibodies, the mnemonic being GAMED, GAMED. अब इनमें क्या क्या story है? Let's just check this out. Okay. Now, every antibody is a monomer. Okay, in this, pretty much every antibody is a monomer, except for IgM and IgA, which exist in some other forms also. I mean, what is their story? Let's just check this out. Here we are. Sirji, IgG, IgE, IgD, and even IgA also looks like this. This is called monomer, one antibody mono. Dimer is IgA. ठीक है, IgA. Doctor साहब, you can imagine 2 different lines as 2 things, and then बीच में एक line जुड़ी हुई है. This bond that holds them together is called disulfide bond. Okay, and there is something called as J chain that holds them together. J for joining chain. Okay, you may forget disulfide bond also, that is also okay. But at least don't forget this J chain. J means join karne wali chain. The chain that joins them is called J chain. So J chain is present in 2 antibodies which exist in multiples. One, it is present in IgM, M for mega or massive antibody. Okay, this is the biggest antibody. Why? Because 5 guys exist as one. So IgM is a pentamer. IgM is a pentamer. This is the first thing. And then IgA, यहाँ पर देख लीजिए, this is that line. You can imagine J chain. A and M. M for massive, biggest, and then IgA. So this part is over, first of all. ठीक है? Now, let's go back to look at the other things. Sir, valency means how many antigens they can bind to. Logically speaking, अगर आप यहाँ देखो, one antibody has 2 binding sites. So IgE, IgD, and IgG, they can bind to only 2 antigens, whereas IgA can bind to 4, 1, 2, 3, 4, and IgM can bind to 10 antigens. So that's it. That's the valency they're talking about. Okay, that's the valency. So you can check this out here. Valency likha hua hai. Now, which is the antibody produced maximum daily? The answer is IgG. IgG. All right, IgG is produced maximum. ठीक है? Next, the one that is having everything maximum is IgG in certain points. So what are the maximum of IgG? He is the winner in all these 3. Half-life, meaning the life of IgG is the highest. The concentration in the blood, serum concentration, is also highest with IgG, and daily it is produced maximum. And the least guy in all the 3, it is none other than your IgE, because IgE can cause allergy. That's the reason it is less. Okay. Next one, moving on, sir. The one which is biggest in size obviously is going to be heavy also. All right, simple logic. The heavy one, molecular weight, is highest with IgM because it is the largest. Because it is the largest, it cannot cross placenta. IgM does not cross placenta because it's the biggest antibody. Massive hai na? Every other antibody can cross placenta, right? Basically, the IgG is the only one that crosses placenta. Why? Because it's the smallest. Okay, I'll write it here. IgG is the smallest, so it crosses placenta. no other antibody crosses placenta. Okay, so excuse me for what I said earlier. No other antibody crosses placenta. IgM तो हरगिज नहीं करेगी. Why? Because IgM is the largest. Other antibodies also don't cross. The only one that crosses placenta is going to be IgG. Next one, sir, opsonization. Who coats up the antigen? Who highlights the antigen saying that this guy is here, come and kill it? Opsonization is done by two, IgM and IgG again. So for most of them, the thing is right here. Okay, I hope this is clear. Now, moving to hypersensitivity topic. Now, this is one thing more or less you can expect one MCQ coming from here. Okay, so hypersensitivity, this is where you got to know something. There are 4 types primarily. Okay, and you're going to remember that mostly the antibodies that are involved is like this. So you're going to remember the anda here, egg. And in the 4th one, there is no antibody. There is no antibody involved. Rather, instead of an antibody, there is cells that are involved here. That's why it's called cell-mediated immunity. That means the damage that is happening to our body, it is due to antibodies in the first 3, but it's due to cells in the 4th one. So 4th one is very different. Okay, 4th one is very different. Now next part, what you got to remember is this word called as acid. Okay, acid. So acid में अब अपने को क्या याद रखना है? The mechanisms that are involved. Sir, the type 1 everybody knows, basically it's called allergy or the word atopy. Atopic dermatitis, atopic rhinitis, they come under this. C for cytotoxic. Okay, C for cytotoxic. 3rd one immune complex, and 4th one is delayed. So this is the word that you have to keep in mind if they ask you the mechanisms. And if you talk about the onsets, after you're exposed to some antigen, how long does it take for your body to produce symptoms? You need to simply understand this. As you keep going down, the time increases. Okay? So in the upper ones, the activation is much faster. Minutes mein ho jata hai upar. Next, as you're going down, the time is increasing. And if you see, I have arranged them in the table in such a format that you have 3 units of time. You have minutes, hours, and days. Okay, so I hope everybody got this idea. Okay. Next, up sir, you got to know type 1. Type 1 mein you must remember the A letters. Mainly asthma, atopic dermatitis, atopic rhinitis, anaphylaxis, drug and food allergies, because they happen in within minutes. You are eating something that you are allergic to. By the time you are not even done with your meal, you get the symptom. तो ये सब किस में आते हैं? Type 1 में. There is one test we do called Cassoni's test for a disease called hydatid cyst disease. Hydatid cyst disease. It's called Cassoni's test, and that is an example of type 1 hypersensitivity. Okay. Now, ये आपको याद रखना होगा that the type 2 reactions are memorized by this mnemonic. My blood group is Rh positive. ठीक है, my blood group is going to be Rh positive. Here we have myasthenia gravis that will be coming into this. Okay, then you have all the remaining ones. ठीक है, मोटा you must remember most of the blood related problems. ठीक है, most of the blood related problems comes in type 2. Type 2. Sir ji, what did I tell you? Type 1 में you have to remember the letter A. First letter is A. Type 2 में the number second one is B. B for blood. Are you understanding this? इसको आसानी से याद रखना. A and B. Done? Moving on. Type 3 में you got to remember this word called sharp mind. Right? In the type 3 you got to remember this, that is sharp mind. Now that we got this idea, sir. Okay. So please check this out. This is some hypersensitivity reactions that you see in leprosy. Mycobacterium lepra reaction. Now for this, I'm just going to share one mnemonic with you. Please remember it like that. The mnemonic is rule of 5. What you have to do is get number 5. लेपरा रिएक्शन में नंबर 2 इज देयर, तो आपको नंबर 2 अगर चाहिए, आई मीन 5 इफ यू वांट टू गेट इन द एंड, व्हाट डू यू डू? 3 + 2 = 5. See, I am just saying that type 2 lepra reaction is an example of which hypersensitivity? Third one, right? It is the type 3 hypersensitivity reaction. लेपरा रिएक्शन 2 टाइप के होते हैं, लेपरा टाइप 1, लेपरा टाइप 2. Each of them is an example of different hypersensitivity. Type 2 lepra reaction is an example of type 3. How are we going to remember 2 + 3 = 5? This is just a mnemonic, sir. There is no logic in this. Next, if you see type 4 hypersensitivity, you have lepra reaction type 1. तो अब 5 कैसे लाएंगे? 1 + 4 = 5. That means the type 4 hypersensitivity, it is nothing but type 1 reaction. And you know this is delayed hypersensitivity. Okay, अब मैं concept में नहीं जाऊंगा. Just remember atopic word were type 1, contact dermatitis is type 4. Atopic dermatitis is type 1, contact dermatitis will be type 4. That means if you are allergic to something related to the skin here, contact dermatitis, you're going to get the reaction 2 to 3 days later. 2 to 3 days later. ठीक है? Next, transplant related immunology. So sir, transplant related pretty straightforward definitions. इसको जल्दी से खत्म कर लेना. Autograft is a graft between same individual. That means it's a transfer that we have done. Transplant we have done within the same individual. I took one part from the body, put it in the other. That's called autograft. Isograft, relatively less thing. Less common. Why? Because it is seen in monozygotic twins. Monozygotic twins. That means there has to be a situation where a transplant is done only between monozygotic twins. Then that's called isograft. Iso means same. Genetically same people. Allograft means same species. Keyword. This is the most common transplant. Why? Because if you are borrowing some organ, you will take it from some person. You're going to take it from some person. Yes or no? So that another guy after HLA matching and everything is going to donate you an organ. So that's it. Most transplants are nothing but allografts. And last and the final is xenograft. Xeno means different species. Xenograft means simply different species. All right. So that's it. Now, कुछ बीमारियां, some diseases related to immunology. First one is B cell defects. You must know that B cells are those cells that produce antibodies. B cells mature to form plasma cells and plasma cell produce antibodies. There is a guy called hyper IgM syndrome. See, सबसे पहले let's revise this concept. Guys, you must know that we have 5 antibodies, but we make the first antibody as IgM. To make other types of antibodies, you need a permission called as class switching. If you want to switch to another type, imagine you need a permission called as class switching. So when you are infected with something, your body will quickly make IgM. That is the reason when you're recently infected, the antibody we detect in some patient is IgM antibody. IgM is the first antibody to be formed. After some time, IgM will disappear and the other antibodies will appear. So for this process to happen, some permission is needed called class switching. And in that class switching, there is one step that is required. What is this step? The step is this. Sir, CD40 has to interact with CD40 ligand. Okay, it is an interaction that is happening between two cells. One cell that is producing the antibody called B cell. Then the next one is T cells, right? B and the T, they talk to each other to produce different antibodies. So agar this step is happening, you can produce other antibodies. If this step is not happening, then you cannot make other antibodies. You will be stuck with one, that is IgM. Okay, you will be stuck with only IgM antibody and it will increase. That's why it's called hyper IgM syndrome. Hyper IgM syndrome. So ठीक है, now that we are done, let's just remember this. Hyper IgM syndrome is a defect of which step, sir? CD40 ligand. That's all it is. Next, you must have learned DiGeorge syndrome. DiGeorge syndrome is a defect in the chromosome 22. But if you go anatomically, this is a defect in third and fourth pharyngeal pouch. Okay, third and fourth pharyngeal pouch. And if there is a defect in the third and fourth pharyngeal pouch, at least you must remember two main organs will not develop. One is thymus, another is going to be your parathyroid gland. Parathyroid gland. Thymus and parathyroid gland do not develop. Sir, if thymus is not there, T cells don't develop. That's why we are learning it in the T cell defects. And you have to remember that parath hormone is not there, hypocalcemia will happen. So hypocalcemia and infections regularly should hint you towards DiGeorge syndrome. Like that, दूसरे भी ये सब diseases हैं इनके अंदर. Like for example, severe combined immunodeficiency. Combined means B and T cell defects. Okay, these are the things that will be taught to you in pathology. These are the defects in certain genes right here. Viscott-Aldrich syndrome. Once again, this is a defect in the B and T. Chronic granulomatous disease. This is a defect in one enzyme called NADPH oxidase. NADPH oxidase. When you are having defect of this, you cannot do the phagocytosis easily. You cannot kill the infections easily. Especially in this people, the infections from certain organisms called catalase positive organisms will happen. Keep that in mind, please. Okay? In the CGD disease, chronic granulomatous disease, you must remember there's a defect of NADPH oxidase, and then they get infections from which type of organisms? Those organisms that has one enzyme called catalase. Okay? Shediac-Higashi syndrome. You must have studied in path. It's called List gene defect. LAD, another disease. So ये सब ना इन पे एक बार put your eyes through the topic of pathology. These are all quite important diseases. ठीक है? Next, starting with the pure micro now, that is bacteriology. So what I have done, as you can see, to save time not to explain 10 things to you, I've just put some things. Cocci means round. Everybody knows by now, cocci का मतलब round होता है. So sir, gram positive organisms are purple in color, gram negatives are pink in color. So if you see somebody who is purple and round, you must know they are gram positive cocci. Examples being staph and strep. Their names itself has coccus, staph and strep. These are the two guys. Okay, clear? Staph will exist in clusters, whereas this guy will exist in chains. Okay, I hope you guys are understanding. One guy is in clusters, grape-like clusters. One is going to be in chains. So that is the story of the staph and the strep. One is in cluster, one is in chain. Up next, what are you going to do to make your life simple is, you just learn gram-positive rods. See, you know already that is staph and strep are the cocci, gram-positive cocci, too basic. अब आप इतना ही करोगे that you're going to learn A, B, C, D, LMN bacteria. Anybody apart from this A, B, C, D, LMN list will belong here. Okay, so you must remember most bacteria will be coming in the gram-negative side. The names of bacteria, so many are there. But here gram-positive mein you just have A, B, C, D, LMN type of organisms. So I hope this is clear first of all. And in the gram-negative cocci, iklota Neisseria. Okay, so negative mein it is Neisseria. Gram-negative you have Neisseria. N for negative, N for this guy. Okay, done. तो आपको ये टेबल ढंग से याद रखना है. The better you remember this, the faster you can solve MCQs. Okay, I really hope you guys are okay with this. Next, very important things that I'm going to discuss next. See, imagine you did your gram stain and you found the organism was purple and brown. That means organism is one of the two. It may be staph or it may be strep. It may be staph or it may be strep. So sir, you need to keep in mind, how can I confirm this? I can do one test called catalase test. Catalase test. In the catalase test, the bacteria who has the enzyme catalase, see, whatever the procedure is that you don't need to know now. Just remember the catalase positive organisms will produce bubbles and catalase negative will not produce any bubbles. अब कौन है भाई? Who's going to produce catalase? Just now I told you catalase positive organisms will generally cause diseases in CGD. So this guy is staph. Staph is catalase positive, whereas strep is catalase negative. So if you see bubbles, you know it is staph. If you don't see bubbles, you know it is strep. पहले तो ये पता चल गया. Now imagine we saw the staph. अब आगे क्या करना है? I need to know the species of staph, right? We are going to remember that staph की कौन सी species है? Two things. If you see bubbles, that was catalase test. But now if you're seeing clots, clumps forming, right? Clumps forming, this is a test called coagulase test. Coagulase test. Coagulase में coagulation दिखेगा. You're going to see some clumps forming. Sir, if you see the clumps, that's called coagulase positive. If you don't see the clumps, that is called coagulase negative. Who's the guy who's going to form clumps? The answer is Staph aureus. Staph aureus. The guys who are not going to form clumps, they are called cons. पहले इसको अच्छे से रट लेते हैं. What is cons? Nicely named. Coagulase test. Coagulase का CO. They are negative. So the negative is N for N here. Then there are staphylococci. Con stands for coagulase negative staphylococci. And there are two guys that come under this list called epidermidis and saprophyticus. तो चलिए फिर से एक बार करते हैं. Let's do this. First of all, catalase test confirms whether it is staph or strep. You see bubbles, you know it is staph. You don't see bubbles, it is strep. ठीक है sir. Now that you know it is staph, I need to know the species. Coagulase test tells me the species. If you're seeing clumps, it's called coagulase positive, it is aureus. If you don't see the clumps, they are together called as cons. And कौन आते हैं आपके cons में? Epidermidis and saprophyticus. Okay, done. Next, अगर strep निकला तो कहानी बदलेगी. What if you didn't see bubbles? If you didn't see bubbles, then it is strep. अब how to know the species of strep? Okay, how to know the species of strep? Now please have a look here. Doc Saab, you put this organisms on blood agar. You will be seeing that they will break down the RBCs. Breaking down RBCs is called hemolysis. So the streptococci can show three patterns. I'm discussing the two main here. One is called beta hemolysis, another is called alpha hemolysis. In the beta hemolysis, yellow color is seen because of the release of bilirubin. Bilirubin imparts yellow color. Bilirubin is going to produce the yellow color. Alpha hemolysis is greenish, dirty greenish color. You can imagine this is due to biliverdin. Okay. Now, who shows this yellow? Who shows this greenish color? That's what you're seeing, dirty green, right? So let's see here. Dr. Saab, if you see, first of all, beta hemolysis, there are two organisms, one called group A streptococci, another called group B streptococci. Group A and group B. Group A is also called as Streptococcus pyogenes. Okay, Streptococcus pyogenes. And group B strep is also known as Streptococcus agalactiae. ठीक है? Agalactiae. So first of all, now that you got this here, group A strep are also called pyogenes and group B strep is called, agalactiae. Now that we know this, इनके कुछ और भी टेस्ट हैं that I want you to memorize right here, पहचानने के, how to recognize them. See guys, as I told you, this is where I'm not going to talk about details. अपने को बस एग्जाम की हद तक पढ़ना है. PYR positive is pyogenes. Pyogenes is PYR positive. Another guy who is PYR test positive is Enterococcus. Doctor Saab, micro ke questions come like this only. They will put some hint and you have to pick up the answer. Okay, first thing done. Next, you must remember that A. galactiae are group B streptococci. And for them, there is one important test called CAMP test. CAMP test. In this CAMP test, you need to see arrow shape that I'm going to talk about later on. Okay? Group B strep mein CAMP test is positive. Once again, the same reminder, you're going to a camp, you're going to take arrows and dart. Campylobacter, camp yaad aaya? Campylobacter shows that, darting motility, and camping, if you go to, once again, you have to remember arrows you will take. So this is over first of all. Next, alpha hemolysis, there are two organisms, one called Streptococcus pneumoniae and Streptococcus viridans. Streptococcus viridans. I hope you are all able to understand this. Alpha hemolysis is this. Okay. इन दोनों को हम एक एंटीबायोटिक यूज करते हैं ऑप्टोचिन, बाय विच वी आर गोइंग टू डिफरेंशिएट देम. ऑप्टोचिन, ठीक है, POV बोलते हैं ना, पॉइंट ऑफ व्यू. हर एक का अपना अपना पॉइंट ऑफ व्यू होता है. सो POV, प्लीज रिमेंबर दिस. ओके, सो डॉक्टर साहब, व्हेन यू आर टॉकिंग अबाउट दिस वर्ड antibiotic you're using, so that means one bacteria may die, one bacteria may not die. That's how we differentiate it. One survives, one dies. Pneumococcus, if you are talking about optochin antibiotic, this bacteria will die if you use it in the lab. That means I will know this is pneumococcus. But if the bacteria doesn't die, resistant word is used. Resistant is viridans. Okay, resistant is viridans. Optochin agar aap use karoge in the lab to figure out whether it's pneumonia or viridans, the guy who dies is called sensitive. Pneumococcus is sensitive and the other one is resistant. Okay, now that's all. Let's move. You must remember that Streptococcus pyogenes, it causes sore throat and skin and soft tissue infections that are being mentioned here. Following this, some people may develop some hypersensitivity reactions, and once they develop hypersensitivity reactions, they will develop diseases like this. One is called rheumatic fever, another is called PSGN. That means सबसे पहले आपको क्या होगा? First of all, you're going to get these problems, either sore throat or the skin infection. Later, you're going to get these things called RF or PSGN. RF or PSGN. Look at the first two letters of PSGN, post streptococcal. That means it happens after streptococcal infection. All right. So they will mention some kind of a respiratory infection. Then they will shift the question towards this kind of things. Rheumatic fever, something for which you have learned the Jones criteria. Yaad hai? Jones criteria, rheumatic fever. So who's the bacteria associated with that? Group A strep. Whenever you study internal medicine, you will learn rheumatic fever always have history of sore throat in the past. Okay, वैसे ही PSGN. What is special with PSGN? You got to know. You must remember that smoky cocoa colored or cola colored urine. Okay, cola colored urine, dark urine, because it's a nephritic syndrome. And I told you already, like rheumatic fever comes under type 2. My blood group is Rh positive, R for rheumatic fever. And if you see sharp mind, type 3 is PSGN. ठीक है, sharp mind, PSGN. Okay, so ये complications है दोस्तों. These are going to happen much later. Done. Next one. Bacillus anthracis. Doctor Saab, this is a guy that has a capsule made up of protein. The only bacteria to have a protein capsule is Bacillus anthracis. Remaining bacteria, they all have sugar capsules. Okay, sugar capsules. This is the only guy to have a protein capsule, Bacillus anthracis. Sir, there are two types of anthrax, one called cutaneous anthrax, another called as pulmonary anthrax. So in the cutaneous anthrax, you're going to get a eschar. There is a black necrotic area that you're going to get called as eschar. And because it's a skin disease, you can remember it is seen in people who are transporting skin. And what is the name for that reason? Hide porters. Porter means transporting something. What is he transporting? Skin of animals. Anthrax comes through cattle, cows, etc. If you're taking their skin for leather, you're going to get this disease. Okay. Next, second thing, pulmonary anthrax. This is the one that is fatal because you are inhaling the spores of anthrax that goes to your lungs, damages them. This is why it's called wool sorter's disease. So that means both of these diseases are coming in some kind of profession. So one is called hide porter's disease, another is called wool sorter's disease. Done? Next one, Dr. Saab, how to identify this bacteria called as Bacillus anthracis. Sir, number one, it is the only guy who has a protein capsule. So protein capsule is detected by a test called McFadden's reaction. McFadden's reaction. All right. So this is the first thing that you must know. ठीक है. अपन sir, I know it's a stretch of things, but कुछ चीजें ऐसे ही याद आ जाती है. When we go to McDonald's, we eat burger. ठीक है. So we are going to remember B for burger, that is McDonald's. McFadden reaction is only for Bacillus anthracis. Bacillus anthracis. Okay. Now, if you talk about other capsules, we have a test called Quellung's reaction. Quellung's reaction is for other capsules called sugar capsules, whereas for the protein capsule, you have McFadden's. Next, we are talking about bacilli. Bacilli are rod-shaped organisms. And the way Bacillus anthracis arranges itself, it looks like a bamboo stick. This is why it's called as bamboo stick appearance. Bamboo stick appearance. Okay? Now imagine that Medusa, aapko yaad hai? Medusa was a, this mythical character. Basically, it is said that she has head. The hair looks like snakes. Okay? Kya karenge? She is having snakes on her head. Okay? अगर समझो this woman Medusa appears in front of you, the snakes are coming towards you. Don't you want to hit her on her head only? Imagine we are taking this bamboo stick and uske sar pe marna hai. Now you want to hit her on her head. Medusa head colonies. Medusa head colonies are once again this. Okay? Now we have killed her. Let's celebrate. Christmas aa gayi. Inverted Christmas tree appearance. Okay, this is how it's going to look like. Inverted Christmas tree appearance. So I hope you guys are understanding this. We are going to relate and remember all these three things. Bamboo stick say you're going to hit on the Medusa's head and then celebrate after that, right? That is where Christmas comes in. So you have to remember this is Christmas tree. Okay, so that's all is the story about this. Next is Bacillus cereus. All right, Bacillus cereus, it seriously causes food poisoning. Bacillus cereus seriously causes food poisoning. And what does it do? It makes you vomit. Okay, it makes you vomit. All right, it is actually copying another type of a bacteria in this, and that bacteria is called Staph aureus. Staph aureus. Technically, it is copying that Staph aureus. Now you can think about it like this. Staph aureus is the superstar of microbiology. So heating the toxin of this guy is not going to damage it. It is heat stable. Staph aureus ke properties hai. What are the properties of Staph aureus toxin? One is it is heat stable. Second, it is fast, very fast in fact. Okay, so you got to remember that Bacillus cereus is copying this guy called aureus. So 3-4 times you got to say this together. Aureus cereus, aureus cereus, aureus cereus. These are the fast food poisonings. And you can see they are heat stable. Chinese fried rice. Chinese fried rice. Sir, fried rice means they are heating the food. Heating the food is also not killing the toxin. That's why it is called heat stable. Heat stable. Okay, so you need to keep this in mind, please. Reheated foods like Chinese fried rice. Chinese fast food poisoning आएगा, so you must choose Bacillus cereus as the answer. All right. So now that we have gotten this, next, Clostridiums I've already discussed. One was tetany, one was botulinum, remember, one contracts your muscles, one relaxes your muscle. Now the remaining two, one is perfringens, one is difficile. So perfringens, you need to remember, this guy is very dangerous. It is the guy that is going to cause gas gangrene, okay, gas gangrene. So you must remember this. This is the guy causing gas gangrene. Fine, this is the first thing. This was previously called Welshi also. Now, to recognize, there are two important reactions, one called Naegler's reaction, images are coming after some time, and another called target hemolysis. Okay, so target hemolysis. So ये दो चीजें आप कम से कम याद रख लेना when it comes to perfringens. Although there are many other reactions that we need to remember, at least I am telling you these two things. One is Naegler's reaction, another is target hemolysis, and in fact one more we can write down here, that is reverse CAMP test, positive. This is the guy in association. The reason reverse word comes here because of the way the procedure is performed. Okay, so technically Naegler, target, and this. So we are simply going to remember the recent superstar, okay, recent superstar from the RRR movie, that is NTR. Okay, so if you remember that guy, we can easily remember these three things. NTR yaad rakhna hai. Okay, NTR. So imagine that scene, right, where he is in front of the tiger. Let's say tiger has scratched this guy and he got a gas gangrene now. ठीक है, टाइगर के सीन में गैस गैंग्रीन किसको हुआ? एनटीआर को. टाइगर सीन, you are getting this scratching, that is NTR. So Naegler's reaction, target hemolysis, reverse CAMP test. Okay, now that this is over, let's move. Last is Clostridium difficile. This is the guy that is associated with diarrhea. ठीक है, this is what is going to cause diarrhea, called antibiotic associated diarrhea. Using antibiotics is producing diarrhea. ठीक है, using the antibiotics produces diarrhea. First remember that. The most important antibiotics, see, first of all, which are the most common we see these days? Cephalosporins. When you use strong cephalosporins, you may get a diarrhea due to this bacteria entering your body now. That is where, you know, the most common antibiotics are. But in exam, usually they're going to ask you clindamycin. Okay, so please remember, this is going to be very important. Clindamycin. Done. Clindamycin question में दिया हुआ है. Most likely they're asking you this question. Antibiotic associated diarrhea, or another word, pseudomembranous colitis. Okay, so that's all about this pseudomembranous colitis. So basically antibiotics are causing the diarrhea, and ironically some antibiotics will treat that diarrhea. Okay, so you must remember that this is the guy that is causing the diarrhea. Current drug of choice is fidaxomicin, followed by vancomycin, followed by metronidazole. Okay, new drug of choice, most effective drug is fidaxomicin. This is what you got to know here. ठीक है, आ सकता है ये question from pharmacology. So that's it. Some of us are like, I want to know everything. I don't want to miss out on anything. They call it as FOMO, right? Fear of missing out. तो यहाँ पर आपका FOMO खत्म हो गया. These are the three antibiotics they will ask, if at all they will ask. Fidaxomicin, oral vancomycin, metronidazole. ठीक है? So no more fear of missing out here. All right, sir. Acne is due to a dude called Propionobacterium. And in the Propionobacterium, you must know we use topical clindamycin. Clindamycin ointment use करते हैं. All right. Coming to diphtheria, sir. Corynebacterium diphtheria. You must all be knowing that we learnt already. How does the toxin of diphtheria work? Remember the mnemonic CDEF? Yes, Corynebacterium diphtheria works on elongation factor 2. Toxin spreads into the body causing the problems. First thing, you all have seen this picture, definitely pseudomembrane. Imagine a chewing gum stuck into your throat. That's how it looks. That is pseudomembrane. Your neck is going to get swollen up. This is called as bull's neck appearance. ठीक है, अब कहानी स्टार्ट होती है. From here you might have collected a sample from the patient through a swab. Now if you put Albert stain in this, you will be able to see some things called as granules. The picture is coming up soon enough. I'll be discussing once again. So granules will appearance, and here you are going to see this Chinese letter appearance that I will be talking about. अब this is the Albert stain, although there are others like Ponder stain or Neisser stain also. 3 stain. आप नैप या पैन जो भी वर्ड याद रखना चाहते हो, Neisser, Albert and Ponder stain. These are the 3. Okay. Next, they may ask you the growth mediums for diphtheria. One is called LSS, Loeffler serum slope, and potassium tellurite agar. Sir, इतना तो याद होगा आपको, or at least you understand. Selective media is selective for some organisms. That means it's a unique media for that organism. PTA, potassium tellurite agar, is the media for diphtheria. It takes some time to grow the diphtheria, but diphtheria is the only one that will grow on this. This is the best medium. Whereas LSS is fast. What is the advantage of LSS? LSS is fast because the growth is within 8 hours. But which is the best media? Best media is PTA. Okay. 48, 8, 48, 8, 48. ठीक है, टाइम लगेगा पर अच्छा कौन सा है? PTA. And Loeffler serum slope is fast but not the best for diphtheria's growth. Okay. Next, Listeria. Remember I told you it causes tumbling motility. What is Listeria supposed to be known for? Meningitis. It causes meningitis in 2 extremes of age. One is old age, very old age, or in the neonates. ठीक है? Or in the neonates. So this is it. Last and the final, we have Mycobacterium leprae. Okay, we have Mycobacterium leprae, which are also called as Hansen's bacilli. Hansen's bacilli. And you got to remember something, because the name of the bacteria itself is Hansen's bacilli. This dude is, the disease called as leprosy is also called as Hansen's disease. Leprosy is also known as Hansen's disease. Dr. Saab, once you develop this disease, you got to remember that depending on how your immune system is, you will develop all these different types of leprosies. Okay. See, broadly speaking, there are 2 types of leprosy. One is called tuberculoid leprosy. Tuberculoid leprosy. Another is called lepromatous leprosy. Tuberculoid leprosy and lepromatous. If your immune system is decent enough, meaning strong, the disease is going to be mild. The mild disease of leprosy is called tuberculoid leprosy. Acha hai, red, green one. The dangerous one is lepromatous. Lepromatous is the dangerous form of leprosy. And on what thing are these things dependent? Immunity. Sir, imagine your immune system, right? Imagine your immunity here. If your immunity is very strong, you will develop tuberculoid leprosy, which is called TT. Okay, this is the one that is represented by the letters called TT over here. Next, if your immunity is weak, you're going to develop something called lepromatous leprosy. Lepromatous leprosy is the bad one, 2 extremes. And exactly in between them, there is a center that is called borderline leprosy. Borderline leprosy. So these are the things that you need to know over here. Now, type of lepra reaction, I have already talked about this, sir. I have talked about 1 plus 4 is equal to 5, 2 plus 3 is equal to 5. That's how you're going to remember the lepra reactions. Now, Doctor, start looking at this. You're seeing a arrow. You're seeing a arrow. What test do you think this is? CAMP test. And CAMP test recognizes which bacteria? It is group B strep, also called A. galactiae. Okay? A. galactiae. So basically this is A. galactiae. Okay. Next, if you don't see the arrow, this is pyogenes. This one is pyogenes. This one is agalactiae. All right. Agalactiae is the most common cause of neonatal meningitis worldwide. Okay, neonatal meningitis, it is caused by this. Okay. Next, if you see here, you're seeing round organisms which are in couples like this. Round organisms, purple color, meaning gram-positive cocci, but they are in pairs. This is Streptococcus pneumoniae. Okay, Streptococcus pneumoniae, because they exist in pairs. We call them as diplococci. Di means two. Doc Saab, this is the bamboo stick appearance. Bamboo stick. You can see line by line, one rod-shaped organism. This is nothing but this. Okay, your bamboo stick. This is your diphtheria. Diphtheria ki pseudo membrane. This is the diphtheria on the gram stain. Purple color rods. Gram stain. They are said to have cuneiform appearance. They arrange very randomly like this. You know. So this gives them this kind of appearance called Chinese letter pattern. Chinese letter pattern. Okay, V या L shape में ऐसे आते हैं. That is where they get this Chinese letter pattern. This greenish color one, this is what is called as Albert stain. Okay, this is what is called as Albert stain. Because they are having appearances like this, that's why they are said to have Chinese letter pattern. And at the ends you will be able to see this granules, metachromatic granules. Okay, last and the final black color colonies is on this PTA agar, potassium tellurite agar. This is diphtheria. Okay, this is diphtheria. All right, so I really hope everybody got this idea right here. So technically done with this, sir. These images above yahan pe. You can see on the blood agar there are some target like appearances. See, whenever you play dart or something, the center part is always having a different color, dart. So you can see this is what is called target hemolysis, NTR. Nagler's reaction, this is your Nagler's reaction. Okay, T for target hemolysis and the reverse camp test. I hope everybody got this. And last and the final, you're looking at acid-fast stain. Okay, you're looking at the acid-fast stain. Doc Saab, in the acid-fast stain, the background looks all cloudy. And against the background, you will see this deep pink color. It's not a baby pink color, deep pink, fuchsia color. That's why in the acid-fast stain, the color is called carbal fuchsia, dark pink. All right. So this is nothing but your TB. These are rod-shaped organisms appearing like this. Cloudy background is acid-fast. Bacilli, rod-shaped organisms, it is TB. So this is where you're looking at the TB organisms. All right. So done with this, gentlemen. Now we are switching to the next aspect, that is Neisseria meningitidis. The name itself tells you that I am the guy who caused meningitis. Although the name is saying I cause meningitis, it's not the most common cause. Please mind it. The most common organism causing meningitis is Streptococcus pneumoniae. Streptococcus pneumoniae is the most common organism causing meningitis. It's not Neisseria. Okay. Now, Sirji, you must know that out of all the organisms that causes meningitis, Neisseria meningitis is one of the organism who gives you the hint I cause meningitis. Sir, Streptococcus pneumonia also cause meningitis. Listeria monocytogenes, it may also cause meningitis. Haemophilus influenza, it may also cause meningitis. All of them are causing meningitis, but none of them have meningitis in the name. The only guy having meningitis in the name is Neisseria meningitis. So, sir, let's give it some respect. Kuch to khas hoga uske paas. Rash and shock. So if they are making a clinical case scenario, apart from the meningitis, they are mentioning rash or a shock, please know they are talking about Neisseria meningitidis. And here we go. The important complication, because there are a couple of things involved in this. They have made a syndrome name here called Waterhouse-Friderichsen syndrome. Waterhouse-Friderichsen syndrome. Okay, I hope this is clear. Next, Neisseria gonorrhoeae. It is a STD, as you all know. Okay, it is a STD. So it presents as a discharge. Okay, it's not a lesion. You can't see anything on the genitalia usually. It is a discharge. Now, in males, it presents with a discharge called urethritis. Through the urethra, there is a discharge that will come out through the genitalia. Same way in the females, they get cervicitis. And long term, they may get something called pelvic inflammatory disease. And lastly, in the neonates, it causes conjunctivitis. Everything is a discharge only. And ditto, everything that you're seeing here, it is seen by somebody called chlamydia. So Neisseria and chlamydia, they both are going to do the same things. And which is this chlamydia? Chlamydia trachomatis. Chlamydia trachomatis. It's going to do pretty much the same things that you're studying with gonorrhea. Okay, done. So they both presents with discharge, and chlamydia is quite a popular STD. All right, done. Next is Haemophilus influenza. Okay, Haemophilus influenza. Now, you have to remember this thing from its name itself, that this guy is indicating that it loves blood. Okay, it has declared that I love blood. Now, although it says I love blood, it likes only couple of things from that. When the RBCs break down, that is after hemolysis, there are some factors called factor 5 and factor 10 being released. And these are crucial for the growth of Haemophilus. Haemophilus ko kya chahiye? Factor 5 and factor 10. Okay. So here you must understand factor 5 and factor 10 are exactly required for the growth of Haemophilus. And guys, there are two culture media which contains blood. Okay, there are two culture media who has blood here. One is called blood agar, logic. Second is chocolate agar. Chocolate is called chocolate, but it's not chocolate. It is hemolyzed blood. Hemolyzed blood. Okay, hemolyzed blood. So sir, simply we are talking about love, right? If you are claiming that you love someone, there are two ways. One is direct way, you will say I love you. That is chocolate agar. It's already hemolyzed blood. It contains this. Or otherwise, आप घुमा फिरा के बोलोगे. Yes or no? You will not say the I love you directly. You're just going to do the things indirectly and convey that you love the other person. That is blood agar. Blood agar is unhemolyzed. Blood agar is unhemolyzed. So technically you cannot grow Haemophilus on this. Haemophilus has already said that it wants factor 5 and factor 10. Okay? It's not like us human beings. Human beings are singing, "Tu hai to mujhe phir aur kya chahiye." That means you are saying you don't want anything. You just want that girl or that person. But that's not Haemophilus. Haemophilus is straightforward. Usko jo chahiye, he's saying. He is claiming I want factor 5, factor 10. Okay. So that is only available on chocolate agar. So if you want to grow this guy, directly you can grow on chocolate agar. But on blood agar, you can't grow it. Because blood agar is unhemolyzed. Blood agar is unhemolyzed. So what can I do to grow it? Indirectly, you have to do something now. You will take blood agar. You will take blood agar. On the blood agar, you will add a bacteria called Staph aureus. And what will Staph aureus do? It will break down RBCs. It does something called beta hemolysis. In other words, now that place where Staph aureus is present, it has broken the RBCs. It has converted to chocolate agar in that area. So blood agar pe directly I cannot grow. So I will put Staph aureus as a mediator. Old love stories. What used to happen in the 80s, 90s? Directly the person doesn't go and say I love you. Friends they will send. Friends जाके convey करते थे I love you. In today's era, you can't do that. Friend harami nikla to wo leke nikal lega. So that's the problem. Okay. So this is where it is. Now you understand, one is chocolate agar, one is blood agar. One is direct, one is indirect. Okay. Now, all this being over, I hope you understood the concept of satellism here. Satellite means the Haemophilus grows around the aureus because the aureus has created hemolysis. Okay. Next, Haemophilus ducreyi. Haemophilus ducreyi, it causes one STD by the name of chancroid. Doc Saab, chancroid ka D you're going to remember with ducreyi. D for ducreyi, chancroid. And the word is ducreyi if you want to pronounce it better, because it is painful lesion. Haemophilus ducreyi, chancroid, D for ducreyi. That is where the mnemonic is do cry. Why? Because it is painful. All right. Next, you have somebody, Treponema pallidum. Treponema pallidum, that produces a STD by the name of syphilis. Treponema pallidum produces syphilis. So, Dr. Saab, syphilis, you must understand, first stage of syphilis, first time you get it, it's called primary syphilis. It's called chancre, which is painless. Okay. Same, chancroid, chancre looks similar. The only thing is one is painful, one is painless. Painful, how to remember? Do cry. You also remember the organism by that mnemonic. Third, secondary syphilis, presence with condyloma lata. Now these are multiple lesions that are spreading out in that genital area. So condyloma lata, you must know how to differentiate it from condyloma acuminata. Acuminata is seen by a virus called HPV, cauliflower-like lesions. So in derma, you must have been told that acuminata versus lata, there are some differences that you must know from derma. Okay. Third, tertiary stage of syphilis. Now this time it's going to involve the CNS and the CVS, the forms of which are written over here. Okay. This is all it is. Next, when you want to diagnose this dude called syphilis, okay, when we are talking about the diagnostic aspects over here, you must keep in mind something. It cannot be gram stained, so you're not going to use your routine microscope called light microscope or light field microscope. So we are going to use something special called dark field microscope. So this is that microscope that creates a dark color background. Okay, dark field microscopy means dark background. So this is all it is. So you will see the organism against this dark background, and it's going to show this spiral motility called as corkscrew motility. Okay, corkscrew motility. ये टेस्ट के नाम याद रख लेना. The screening tests for syphilis are called VDRL and RPR. Okay, and the confirmatory tests, they are having the letter T in their name. See, we are talking T for Treponema pallidum, so इनके नाम में T आता है. Actually, that T stands for treponem. For example, TPHA stands for Treponema pallidum heme agglutination test, Treponema pallidum immobilization test. So that full forms are not needed. You just need to remember T for Treponema. T दिख गया, it's a confirmatory test. नहीं दिखा, like VDRL and RPR, you will say it's a screening test. And you must have all learned that usually if your examiner wants to, let's say, ask you about the STDs, they may mention the word truck drivers. Okay, that stereotypical thing they're going to mention is truck drivers. Okay. Next, Leptospira. Leptospira, last one, you have to remember this is the guy that is going to be from the rat's urine. It causes a disease called as leptospirosis, which is also called by the name of Weil's disease. Okay, Weil's disease is by leptospira. Answer एक mnemonic है that you must remember, that is cat sat on mat. ठीक है, cat sat on mat, maybe later. Okay, cat sat on mat, maybe later. So what is this cat sat on mat maybe later? Let's remember this mnemonic. C for cold agglutination test, ये तो same रहेगा. AT stands for agglutination test. ठीक है, the only words that will change is C, S and M. C for cold agglutination test, S for standard agglutination test, and M for microscopic agglutination test. Maybe later, M for mycoplasma. That means cold agglutination test is done for mycoplasma. Standard agglutination test is done for brucella. And third one, MAT is done for leptospira. That's it. So you're going to remember this mnemonic. Cat sat on mat maybe later. Okay, so that's it. Next, there is a gentleman called by the name of Rickettsia, as you all know. Now, one of the most famous diseases of Rickettsia that we see in India, it is called as scrub typhus. It is going to present with a eschar. Doc Saab, in your exam, if you see a eschar, only two diseases you must think mainly if it's a micro question. Either it is anthrax, we studied already, and second one is Rickettsia that you're looking at right now. Which one? Scrub typhus. Scrub typhus. Or usually इसकी drug of choice भी important होती है doxycycline, right? For the rickettsial infection, doxycycline is the drug. Fine. Next, moving further to E. coli. Dr. Saab, you need to know that E. coli is the guy that is the most common cause of UTI. And every statement I've written over there, any age, male, female, community acquired or nosocomial, hospital acquired also, UTI, it is E. coli that is more common. And as a matter of fact, usually when you put catheters in somebody, they get UTIs. So it's called catheter associated UTI. So for cocci also, the organism is E. coli only. Catheter associated UTI also, E. coli is the guy. Now, apart from this, E. coli is also associated with diarrheas. Now, there are two main diarrheal strains that I want you to know. One is E-tech, another is E-hec. Sir, when you talk E-tech, T for T, you're going to remember traveler's diarrhea. And traveler's diarrhea is a watery diarrhea. And you should recall when we learned watery diarrhea, what did I teach you earlier? I told you something back then. I told you that if you're talking watery diarrhea, then the organism must be increasing a thing called as cyclic AMP. Cyclic AMP. All right. Next, enterohemorrhagic E. coli. This guy, the name itself says it's going to produce a bloody diarrhea. And I told you that it works by damaging our 60S ribosome. 60S ribosome. All right, that will stop the protein synthesis. Okay, that's your E. coli. Next, Klebsiella. Doctor साहब, club pronounce करते हैं इसको Klebsiella, Klebsiella. When you go to club, okay, you want to see people in red lipsticks. Okay, you want to see everybody, having red lipsticks or something, obviously. So, sir, Klebsiella mein everything you have to remember with red. If you talk pneumonia, this pneumonia is such that the person is going to have a sputum which is bloody red. Okay, it is having blood in it, right? Red blood type of a thing. And if you see the other guy in this story, that is STD, Klebsiella granulomatous, it causes a STD. Now, this one is presenting with beefy red ulcer. Beefy red ulcer. Doctor Saab, if you go to club, obviously this you are going to do. So, this pneumonia is usually seen in alcoholics. तो याद रखना है अच्छे से, right? You have to remember this club. If you're going to a club, you want to see people in red. And as I said, इसमें red दिखेगा हर चीज. Alcoholic people, you're going to a club to drink. Second, if things goes wrong, you may get some bad news. As you're seeing here, there is a STD called donovanosis. ठीक है, डोनोवनोसिस. ठीक है, जब आप दोनों मिलेंगे तो क्या हो जाएगा भाई? red color ka ulcer. On the genitalia you will see this beefy red ulcer, beefy red ulcer. Now when you see this agent, it will be usually seen inside a macrophage. Let's say this is a macrophage with its nucleus. The organism will be inside this. It's a rod-shaped organism. The organism will be inside, staining at two ends like this. And this is called bipolar staining because it's staining at two ends, or it is also called safety pin appearance. It is also called by the name of safety pin appearance. Okay? So this is all that you got to know. Next, Legionella. Okay, Legionella. Now, Legionella is the guy that will come with contaminated air conditioners. Contaminated air conditioners. And sir, what is going to happen is, obviously, if it's coming through air conditioner, you're inhaling something, you will get a pulmonary disease, right? It's a pneumonia. Along with that, they will mention in the history, the patient is also having a diarrhea. Diarrhea. Okay? So Legionella में क्या हो रहा है? Please remember, one is pneumonia plus diarrhea once you get it through the air conditioners. Air conditioners. And the culture media that you got to know for this, it is called buffered charcoal yeast extract agar. ठीक है? नाम देख के ही आपके अंदर कुछ एक फीलिंग आई होगी. Like, you have to understand this. See, we are talking about this. This is for Legionella. है ना? So we are talking about this culture media that is required for the Legionella. So you got to remember, let's say you have something and somebody keeps asking, bhai mujhe dena, bhai mujhe dena. And if somebody keeps irritating you, asking you something over and over again, right? You know, there is one great word that we use, BC, and you'd be like, yele. Yes or no? So you're going to remember it like this, BC, yele. So that is buffered charcoal yeast extract agar, Legionella, Legionella. ठीक है? That is one guy, charcoal है, तो फिर black color का होगा. Charcoal is going to be black color. That's it, it's a black color agar. आराम से याद हो जाएगा अभी. ठीक है, Legionella का है. This is one for the Legionella. Last and the final bacteria, somebody important. In my opinion, this is super high yield guy, Gardnerella. Okay, Gardnerella. This is the guy that is causing the disease called as bacterial vaginosis. Bacterial vaginosis. Okay. Now you need to imagine the movie called Drishyam. You remember that movie that there was a murder that was committed, and then what happened after that is they buried the body in a place they can't even imagine. It's just like that. Okay, imagine here. Okay, there is a garden. Somebody's wife was killed here. Wife को क्या बोलते हैं in Hindi? बीवी. Yes or no? Somebody's wife was killed and was buried in the garden. Somebody's wife was killed and buried in the garden. And Doc Saab, now the thing is, I will get dogs to sniff out whatever is happening. Yes or no? In forensic, obviously the dogs are used. Okay. So here's where you have to remember, there is a test called as whiff test. What are we doing in the whiff test? We are smelling. With whiff, you sniff. Are you understanding? With whiff, you're going to sniff. And what are you going to find? Clue cells. Clue cells. Are you understanding this? With whiff, you sniff. And what do you find? Clue cells. ठीक है? अपने CID में दिखाता है ना, दया कुछ तो गड़बड़ है. So basically you need to find out this thing. All right. So Amsel's criteria is one criteria that is required for the diagnosis. Usually the pH of vagina is acidic, but this guy enters the body when the vaginal pH becomes slightly alkaline. All right. Next, you will get a discharge from the patient. That discharge, if you collect, add 10% potassium hydroxide, it gives a fishy amine odor that is called whiff test, and then clue cells. Okay? And since we are here anyway, let's learn the treatment also for this guy. It is metronidazole. Metronidazole. Okay, this is an antibiotic we use here by the name of metronidazole. Now, what is the mnemonic that I want you to remember here is go get back on metro. Go get back on metro. Now, what is going to be this go get back on metro? Check this out. See, sir, first thing, G for Gardnerella. G for Gardnerella, this is one bacteria. Then three parasites together. What are these three parasites together? Giardia, Entamoeba, and Trichomonas. Trichomonas. All right, I hope you're understanding. And then there is one more bacteria called Bacteroides. Bacteroides is anaerobic bacteria. Anaerobic bacteria. Anaerobes के लिए भी we use metronidazole. Metronidazole. Okay. So go get on metro. Go get back on metro. That's it. Those are the metronidazole, right? That's it. This is the treatment over here that you need to know. Okay. And in this, usually for trichomoniasis, both partners need to be treated, okay, with metronidazole. So done. Now let's look at the pictures. I hope now you're able to analyze the moment you look at this picture. Black color, charcoal like means this is nothing but your BCYE. This is BCYE for Legionella. अब नहीं भूलेंगे, ठीक है? Next, this is where, if you see this first image, this is your Staph aureus, and around the Staph aureus you can see small colonies of Haemophilus influenza growing like a satellite. So this picture is nothing but blood agar, and you're producing satellism here, right? Staph aureus and the growth of Haemophilus influenza. This is your satellism. Now, sir, dark color background is nothing but your dark field microscopy, and you're seeing spiral organism. This is spirochetes. Spirochetes. Okay. Next and the last one, pink color organisms. Pink color organisms, but they're more like comma. They're little bent. Okay. Comma shaped organism. In the school also they teach this thing. This is Vibrio cholera on the gram stain. Okay. Vibrio cholera is the comma shaped organisms. Pink color, they are gram negatives, and that's how they will look. Fine. So that's it. We have discussed all the images here so far. Take it, friends. Now what we will do is we'll just take a 10 minutes breather, and after this we'll get back to the virology section and the remaining aspects. So viruses, fungi, and the parasites, we're just going to do it in some moments. All right. Right, everyone. So welcome back again. Now we just have few more sections left, starting from virology. Guys, सबसे पहले, just like what we did in the bacteria, remember we learned a list called ABCDLMN, and anybody apart from that we had put into the list of gram negatives. Same way, here's where you have to learn the list called happy. Okay, so this list is going to be happy. तो इनको ऐसे ही याद करना है अपने को, HAPPI, ठीक है, happy. Anybody apart from this is going to be a RNA virus. Okay, anybody apart from this is going to be a RNA virus. Clear? Happy अच्छे से रट लेना. Now, once again, the DNA versus the RNA viruses are further segregated into two different varieties. Now, what are these varieties? Naked viruses and envelope viruses. Naked viruses are also called as non-envelope viruses. And these naked viruses, compared to envelope one, they are more resistant, they are more stronger. Okay, so you must remember they are resistant to alcohol, etc. Okay. Now, who are the naked viruses? There are two lists we are going to learn. One list is PAP, PAP. That means these are the DNA viruses that are naked viruses. And then RNA में we have these words, that is CRAP. तो आपको क्या याद रखना है? Naked, PAP and CRAP, PAP and CRAP. So this is the two words that you're going to remember for naked viruses. All right. Now, hepatitis viruses comes in the naked viruses. There are some hepatitis which are naked, some which are enveloped. So, कौन से naked viruses होते हैं? Hepatitis A and E. ठीक है? Hepatitis A and E, they are naked viruses. So, you got to remember them like this. इनके अंदर absent होता है. Envelope is absent in them. A and E, absent envelope. Envelope is absent in A and E. That's it. That's a mnemonic by which you can remember this. Remaining all hepatitis viruses are enveloped viruses. Okay. So this only gives them this resistance powers, etc. That's the reason we are learning this. Naked viruses are more resistant. Okay. Now starting with the virology, the happy list, we have herpes family. In the herpes, the first two members are called HSV1 and HSV2. Okay. Now what you need to remember is, these guys 1 and 2, number 1 is the top number, right? Top number, number 1 is top. So these are the guys that are going to cause lesions in and around the face. They're going to cause lesions in and around the face. So I hope that is clear. Now HSV2 causes the lesions down. Okay, so how are we going to remember them? You are going to take the waist as a reference here, and above the waist is number 1. Number 1 is on the top. Below the waist is number 2. Okay, so below waist and above waist. So this is the location. So this tells us something very clearly now. Below the waist we can expect the genital lesions. Now when you talk about the genital lesions, here's where they are painful and they are recurrent. Okay, they are painful and they are recurrent. That means they keep coming back over and over again. These are the classical ways they're going to describe the herpes for you. I hope everybody got that. Now this guy comes back over and over again. That is what is meant by, recurrence. So if they're coming back again, so they must be hiding somewhere in the body so that they can come back again. That hiding is called latency. Sir, if they are coming in the genital area again and again, so they must be hiding somewhere near the genitalia. So these guys are going to hide in the sacral ganglia, and this guy is going to hide in the trigeminal ganglia. Trigeminal, because this is much closer to your face. Trigeminal ganglia and the sacral ganglia. And last, you can see all the lesions which are in and around the face. Next is, if they want to describe the encephalitis by herpes, mark my words, this is the super important word, temporal lobe encephalitis. Temporal lobe, the moment you hear, you got to know they're talking about HSV1. Okay. Next, HSV2, if you talk about, it is causing lesions below the waist with one exception. That exception is called Mollaret's meningitis. Mollaret's meningitis. All right. Next, after this, for the first three herpes, that is HSV1, 2 and 3, we can do something called Zank smear, which is going to show macrophages which are fused together. You know, in pathology, you call them as multinucleated giant cells. Multinucleated giant cells. So this is all that you got to know right here. Take it done. Next part. Moving further, HHV3 is called as varicella zoster virus. Now this varicella zoster virus, when we are children, it causes chicken pox. As a child, it causes chicken pox, and then later in the adulthood, it gets reactivated. Later, it's going to get reactivated. Okay? Next, what you must know apart from this is chicken pox is a bilateral lesion. That means both sides of the body will be involved. Whereas if you're talking about the shingles, shingles is single-sided. Shingle is single-sided, involving dermatomes. Dermatomes are the nerves of this area. So what happens is the dermatomes run oblique in the body in this area. So the disease rash is also going to come oblique. It's going to come oblique. So you got to remember shingle is single. Now, after you get shingles, you may generally get nerve involvement, and the pain in that, it is called as post-herpetic neuralgia. Post-herpetic neuralgia. All right. Now, next, mark this syndrome. You study this in ENT also, right? Ramsay-Hunt syndrome. This is going to be important. So what all is involved here? Facial nerve is involved, etc. So when you get facial nerves involvement, that can lead to what? Bell's palsy. And there will be some vesicles. Vesicles means fluid-filled lesions that you're going to see on different areas. Okay. So ये था आपका HHV3 varicella zoster. Next, HHV4 is called Epstein-Barr virus. Epstein-Barr virus, which is associated with Kissing's disease. Okay, Epstein-Barr virus is associated with Kissing's disease. So you can remember when you go to a bar, you may end up kissing somebody, right? So Epstein-Barr virus, this is the Kissing's disease guy, infectious mononucleosis. And guys, this organism is also associated with a lot of malignancies, right? And Epstein-Barr virus usually affect the B lymphocytes. B lymphocytes. Okay. So you must remember lymphomas are quite common by this guy for that reason. B cell lymphoma, I mean the Burkitt's lymphoma, Hodgkin's lymphoma, non-Hodgkin's lymphoma, etc. Nasopharyngeal carcinoma, which is quite famous in the Chinese population. Chinese population usually get this nasopharyngeal carcinoma. Next is HHV5, which is called cytomegalovirus. Cytomegalovirus. Mega means big. It has something big that you have to remember. Owl's eye. Owl's eye inclusions. Owl's eye inclusions. That is something you got to know when it comes to cytomegalovirus. One of the most important things. ठीक है sir. Next. So यही owl's eye appearance that you got to remember here. HHV6, number 646, it causes sixth disease. That means in this disorder, the rash presents exactly on the sixth day. The rash presents exactly on the sixth day. That's it. That is your HHV6. Last is HHV8. 8 AIDS में disease करता है called Kaposi sarcoma. 8 AIDS. So this is the guy that causes Kaposi sarcoma in the AIDS patients. Fine. Next is Molluscum contagiosum. This is the guy that produces this dome-shaped lesions. Okay, so molluscum contagiosum में dome shaped lesions होते हैं. McDonald's again. So MCD. So you're going to remember in molluscum contagiosum, you're basically going to get this dome shaped lesions. Now in this dome, exactly in the center, there will be a depression. Imagine you're looking from above, there will be a depression in the center. There will be a depression in the center right here. And when you basically take a sample from there and examine, there is positivity for something called as HP bodies, Henderson-Patterson bodies bolte hain. That's what you're going to see in molluscum contagiosum. Okay. Now that we got this, parvovirus. Another one in the list is parvovirus. Now parvovirus has certain specialities. It's the smallest virus. It comes in the list of PAP, that means it's a naked virus, and it is the only single-stranded DNA virus. All the other viruses are double-stranded except for this one. This is single-stranded DNA. In the list of happy, everybody is double-stranded. This is the only less happy guy, right? This is the only less happy guy. Why is he less happy? He's getting slapped all the time. Okay, this guy is getting slapped all the time. So you can remember the slapped cheek appearance. Those who have attended my classes know we can easily remember we slap somebody by using 5 fingers. That means that 5 you have to remember with the name called fifth disease. Fifth disease. The rash presents exactly on the 5th day. And what is the appearance of rash? Slap cheek appearance. Also keep an eye on the other things. This guy, parvovirus B19, it reduces somebody in the blood. It affects your RBCs. Imagine it's going to reduce your RBCs. Parvovirus B19, it reduces your blood cells. Hence the red cell aplasia. Okay. So these words you need to keep in mind when it comes to parvovirus, main being the slap cheek appearance, 5th disease. Okay. Next, human papillomavirus. Okay, papillomavirus. So this comes when you do some PAP, sexual lesions, right? So when you're doing some PAP, you're going to get this guy, right? So anybody who has sex, imagine this woman gets this virus inside her. She's the one who's very prone to cervical cancer now. Okay, cervical cancer will have HPV virus in the history, obviously. So you got to know something. There are 2 strains, at least what I want you to know as of now. 6 and 11, 16, 18 onwards. 6 and 11, 16, 18 onwards. All right. So this is all I'm saying. Come second. Smaller numbers, a relatively smaller problem. Bigger numbers, a bigger problem. Smaller number is a smaller problem. Bigger number is a bigger problem. That's how we're going to remember. 6 and 11, they come by sex once again. They cause a lesion, genital lesion called condyloma acuminata. I told you this before also. In syphilis, we studied condyloma lata. Here you have condyloma acuminata. Next, doctor, 16, 18 onwards, they cause cancer. CACX means cervical cancer. Now, there are some parts of this virus that helps it to produce the cancer more easily. Sir, this guy knows when it enters the human body, there are some bodyguards we have. Okay. These bodyguards are called tumor suppressor genes or guardians of genome. Are you understanding that? They are called tumor suppressors or guardians of genome. कौन है ये 2 लोग? Who are the bodyguards who protects us from cancers? P53 and RB. They are your friends. P53, RB are the tumor suppressors. This guy is so smart. Human papillomavirus, it knows that when it comes inside to cause a cancer, these guys will stop it. So what is it doing? It's bringing some weapons along with it. It's bringing some weapons. Those weapons are called E6 and E7. E6 will damage the P53 part in us. E7 will damage the RB, and that is going to cause the cancer. Okay, retinoblastoma gene bolte hain na, RB gene. So basically that's it. Next, we're going to talk about the hepatitis. Okay, hepatitis. Now you must remember there were two naked viruses. Naked viruses, who are the naked viruses? A and E. Remember absent, absent envelope, A and E. And you can remember naked. When you're naked, what are you doing? You're defecating. Fecal-oral, they're going to spread by fecal-oral route. Other things, they're going to spread by sexual and other routes, as you can see. Now, if they talk about dangerous hepatitis, you can remember that hepatitis D is the dangerous one. Okay, hepatitis D is the dangerous one. Why is it dangerous? Because hepatitis D, it comes along with another hepatitis virus called hepatitis B. So this is easily remembered like this. Hepatitis D is D for dependent on B. That means they are coming in your body together. Super infection. What is super infection? You will already have hepatitis B inside your body. Then who will join later? Hepatitis D. Okay. Hepatitis D comes later. ठीक है. In Bollywood, there's a dialogue, right? कुत्ते झुंड में आते हैं. Dogs don't come alone. So you can remember hepatitis D, D for dog. It doesn't come alone. It comes with the support of hepatitis B. Hepatitis B. All right. Next, in pregnancy, the most dangerous one is hepatitis E. Normally, if they ask, then you should answer hepatitis D. But if specifically pregnancy is mentioned, then you're going to answer as hepatitis E. Okay, hepatitis E. Bhulna nahi hai, please. D for the dangerous one generally is this, but in pregnancy it is E. Done. Finally, the hepatitis that can become chronic is both B as well as C, but the chances of becoming chronic are more with hepatitis C. C for chronicity. C for chronicity. ठीक है. D for dangerous. Hep C, C for chronic. Hep D, D for dangerous. All right. And the risk is much less in the hepatitis B, okay, compared to this. So that's why we give more weightage to hepatitis C. Done. Now that this part is over, I want you to look at this here, what I'm explaining next. Dr. Saab, hepatitis B, if you talk about hepatitis B, this virus, the original virus, has three parts in it. There is something called HBs antigen, HBe antigen, and HBc antigen. There are three parts that are present in this. As I said, HBc, HBe, HBs. S matlab surface, it's on the top, upper layers of the virus. Now, this is the antigen that allows hepatitis B virus to cause the disease. बीमारी करने वायरस का नाम क्या है? It is HBs. HBs antigen allows it to cause the hepatitis B disease. Now, E for, we can remember, it gives it infectious nature. E for infectious. Now, hepatitis C core antigen is not found in our test, so I'm not going to teach you anything about that. Antigen called HBc is not detected in our tests because it's hidden. Core means it's deep inside. So the only two antigens that we are able to detect by our testing is HBs antigen, HBe antigen. Now anyway, I just explained this to you in the structure. Ye virus bahar tha. This virus is outside. Now imagine this virus came into somebody's body. This is a full virus that came into somebody's body. Now because this virus came into this person's body, their bodies will respond by doing something. Okay, so what are they going to do now is they will make antibodies against each of these antigens. Our body in response is doing what? It's making three separate antibodies. One antibody will be formed against HBs antigen. It's called anti-HBs. One antibody is formed against HBe antigen. It is called anti-HBe. And last and the final, the third one is called anti-HBc. Anti-HBc. So when you are infected with somebody, there will be three antibodies formed. They don't form all at once. Please remember, I'm not saying that all of the antibodies are formed at once. Eventually, you will have all the three. Imagine you got this infection initially. How do you know the infection is present? Virus came inside, and after some time you produced all the three antibodies, and now the disease is gone from your body. But organism jayega, antibodies will remain forever. Are you understanding this? Antigen is gone, organism is gone from the body, but this antibodies will remember. I mean, they will be retained in the body. They will remember the virus and they will stay in the body. Memory cells jaise hum padhte hain. So technically the idea is, if you got an infection in the past, all the three antibodies will be found inside your body. Okay, this was the story when you talk about the natural virus. Fine. Now moving to the next part here. What is the next part? This was the natural virus. Now when we were making a vaccine, sir, we did not take all the virus. We picked only the part which is important. Okay, we picked only the part that is important. So what is the part that is important? Only one antigen, HBs antigen, because that's the guy producing the disease. बीमारी हुई तो फिर आप infectious हो गए and all the remaining things. First the disease must happen, then remaining things matter. So sir, when you take a vaccine, only one part of the virus is entering your body, HBs antigen. So now the story is this: when you take a vaccine, only one antibody will be formed. And what is this one antibody? Only anti-HBs. If you remember previous case I discussed, if you had hepatitis B in the past, all the three antibodies will be there because all the three antigen came in your body. But if you are taking a vaccine, only one antibody will be formed. Why? Because in the vaccine strain, only one antigen is present. Okay, that's it. So now, based on this, we have to see what are the interpretations. And to read this, you have to go in this direction like this. ऐसे पढ़ना होता है. One is positive, one is negative, etc. So here we go. Try to understand this, my friends. See, as long as HBs antigen is positive, that means these people are having disease. Okay, so these three people are having hepatitis B, confirmed. HBs antigen is present, that means disease is present. Now, how will I know which type of disease? Is it acute or chronic? Sir, in immunology today, I told you first antibody is formed called as IgM. IgM represents recent or acute. So if you're finding IgM in somebody, it means acute. पहले तो बीमारी है, yes. Disease present, yes. What kind of disease? Acute. But if you're finding IgG, it means chronic. IgG means chronic. Done. So first case is acute hepatitis, second and third cases are chronic hepatitis. Then we need to also assess, is this more infective, high infective or less infective person? Sir, if HBe antigen is present, E for infectious antigen is present, that means you are infectious. If antibody is present, then you are not infectious. Simple. HBe antigen mila, so you will say patient is infectious, and if it's not present, you're going to say non-infectious. Doctor Saab, first case, let's read again. Disease is present, acute disease is present, infectious. Joining all the words, you can write down acute infectious hepatitis. First case is going to be acute infectious hepatitis. Second case, now you guys can answer on your own, right? Disease is present, chronic and infectious. Chronic infectious hepatitis. Second one is chronic infectious hepatitis. Third one, chronic non-infectious hepatitis. Why? Because this time antibody is there. Chronic non-infectious hepatitis. Then Doc Saab, finally speaking, if you see only one antibody called anti-HBs, that means this is vaccinated person. But if you see all the three antibodies, then you are a recovered person. It's a past infection. You might have had infection in the past. Okay, so this is it. If you did not understand this, please, this video is available on the YouTube. You can watch it again. All right. So I'm going to move forward from here. इसको देख लेना अच्छे से, not so difficult. All you need to do is understand this part I explained, then come here and read this. You can watch it twice at the 2x speed. You should be fine. Question आएगा उस पे more or less. That's why I want you to know this for sure. Next is rabies virus. Once again, a very high yield virus. You must remember, if you're talking about the rabies, this hydrophobia is a very classical feature that you see in this. Sir, next part is this diagnosis, antemortem and postmortem. Antemortem and postmortem diagnosis, meaning one guy has already died. If you're died, what I can do is I can take a sample from your brain now because you're already dead. So that brain will give me something called Negri bodies. Negri bodies, which are usually found in these 3 areas that I have mentioned. And we are going to stain the Negri bodies by this thing called as Seller's stain. Okay, Negri bodies by the Seller's stain. Done. Next part. If you're talking about the person who is alive, meaning antemortem diagnosis, we can take certain samples where the virus is expected. From the back side of the neck, we take the hair follicles, or from the cornea as well. So you can take the samples from here, and then you can do tests like DFAT or PCR. DFAT stands for direct fluorescent antigen test. That means you're detecting one part of the virus called nucleoprotein antigen. Okay. It is done by using an antibody. Anyway, procedure is not important. Just remember DFAT is we are detecting one antigen by the name of nucleoprotein antigen. Okay. Now, coming to the management aspects of the rabies, you got to know that there are 3 categories in which the rabies management would be divided. Category 1 is not having any risk of rabies because you are just touching some animal or licking of the animal happens on the intact skin. If your skin is okay, you don't get the infection. But if your skin is abraded or there is some kind of cut, right, then there is a risk of infection entering inside. So sir, category 1, there is no risk of rabies. I'm not going to give any treatment also. But if you talk about category 2 and category 3, so category 2 is there are small scratches on the body without bleeding. Still there is risk of rabies. So what I'm going to do, I'll clean that area wherever the scratches are present, called as local treatment. You have to clean it with abundant water. You may apply some disinfectant, etc. And just to be on the safe side, we are going to give vaccines so that this person forms his own antibody. Okay, right? Forms his own antibody. Now, next, moving further, category 3, major risk. If there is a person having bites with bleeding, right? Somebody got a dog bite and on that areas you're seeing bleeding. So sir, this is a high risk case because the dog has bitten, the dog has thrown all the saliva into that wound. So obviously this is a high risk case of rabies. Now, when there is a high risk case of rabies, I know that this guy may develop rabies in the future and die. I'm not gonna wait and watch. Apart from cleaning the wound so that I can reduce the viral load in that area, apart from the vaccine, okay, apart from the vaccine, now what you got to do is give ready-made antibodies. These are ready-made prepared antibodies called HRIG. What does the HRIG stands for? Human rabies immunoglobulins. Antibodies are also called as immunoglobulins. Okay, that's it. Now, next part, paramyxoviruses. So, sir, you got to understand this. When you're talking about paramyxoviruses, it's a family which includes all these other viruses like parainfluenza virus. This is the guy that causes croup. Croup is laryngotracheobronchitis. It's inflammation in pretty much all this tract. You can imagine the entire tract is inflamed, because of which you see that inflammation on radiological examination by some sign. I'm sure all of you remember this. It's called by the name of steeple sign. Steeple sign is seen in the croup. Okay? Now, this inflamed area produces the cough that may sound like barking. So this is described as the barking cough. Okay, this is described as barking cough. With mumps, you got to know parotitis is a very important thing, that is inflammation of the parotids. But your exam has multiple times asked orchitis, inflammation of testis. Inflammation of testis has been asked when it comes to the mumps. So from all this family, measles is the highest yield guy. What happens is when measles begins, the symptoms has a procedure like appearance, like one after the other, it's going to present. So first of all, when you get measles, the symptoms begin with something called prodromal phase, where the person gets fever, any infection, plus something what we call as 3 C's of measles: cough, coryza, conjunctivitis. Coryza means runny nose. Okay, cough, coryza, conjunctivitis. Next, exactly 2 days later, 2 days after the prodrome, the patient is going to present with Koplik spots. Okay, this is happening exactly 2 days after the prodrome phase. Koplik spots are the unique thing that you find in measles, which are in the buccal mucosa, right? And then exactly 2 more days later, that is 4 days later, the rash will appear. Okay, 4 days later. This I'm talking not from incubation period, I'm talking from onset of symptoms. जब से symptoms start हो गए, so 4 days later you're going to see the rash appearing in the measles. Later you may get these complications that are there like otitis media, etc. Usually your exam would love to ask this rare complication which is fatal because it's serious. They may ask you this. It is called as SSPE, subsclerosing panencephalitis. Subsclerosing panencephalitis. Okay. Next, there is a fancy name given to the macrophages here called Warthin-Finkeldy cells. Zank cells we studied in herpes, like that Warthin-Finkeldy cells are here. Okay. Next, rubella, German measles. Now you must know that this dude is from that torch. ठीक है, torch. Imagine आजकल special someone आपके आसानी से नहीं मिलते. Imagine you're searching your special someone by using a torch and you found that beautiful lady. Beautiful lady is called Bella. Bella, right? If you remember from my classes, I used to say that when you fall in love with someone, you kind of become blind, right? You're blinded. Now you don't see anything else. You don't hear other people. You become deaf. So you got to remember that Bella means a beautiful lady. So you're blind and deaf in the love of rubella. Love is your heart, PDA, patent ductus arteriosus, blind and deaf, blind and deaf. So this guy is gonna make you blind and deaf. So blind and deaf in the love of rubella. Okay, in Hindi we used to say प्यार में अंधे बहरे, याद है? So that's how we used to remember rubella. Done. Next, moving further, dengue fever, the virus called by the name of dengue virus. So dengue fever virus. So earlier there used to be 4 varieties of dengue. Now a 5th one also has been identified. Anyways, that doesn't matter as much. Aedes mosquito is spreading this. Majority of the infections are spread by Aedes mosquito only. Culex spread main 2. कौन से 2 है? Japanese encephalitis and filariasis. Japanese encephalitis and filariasis, these are spread by Culex. Okay, so this you got to know. Chaliye bhai. Ab talking about this dengue fever has something special called as antibody dependent enhancement. So what is this condition? Let's check. See, imagine you live in some area where there are a lot of mosquitoes. Okay, so what happens is you might get infected with some mosquitoes first time. That is called as primary dengue infection. Let's say that you got infected by den 1 for the first time, right? Just in a sequence, den 1 out of the 5 you got infected by 1. Now your body will make antibody towards this virus. Okay, so you are pretty much protected from den 1 variety starting from now because you already got infected. अब आपको protection रहेगी against den 1, not against the other varieties. So now what happens when a new virus, new strain enters your body? Let's say den 2 enters your body. Den 2 is supposed to be the most dangerous. Okay, den 2 variety is the most dangerous variety. Now this older antibody doesn't kill den 2. It doesn't kill den 2. It doesn't do, anything helpful for us. Rather, our own antibody ditches us and it goes and protects the virus. Our own antibodies, our own immune system, it's like protecting the enemy. Now the enemy gets more better chances to multiply and damage your body, causing reduction in the platelets. Because there is reduction in the platelets, it leads to bleeding that happens inside the body. That is why you get this presentation called dengue hemorrhagic fever. Hemorrhagic. Platelets usually are less than 1 lakh here. Then what happens is, if you don't control this bleeding, it leads to the next sequelae called dengue shock syndrome. Dengue hemorrhagic fever followed by dengue shock syndrome. Why? Because platelets are less. Now, who's supposed to be blamed for all this? Our antibodies. They enhance this disease. Hence, this concept is called antibody dependent enhancement. Antibody dependent enhancement. Okay. So now that we have understood this, let's look at this part, sir. Now diagnosis. Please remember, when any infection comes in the body, the organism comes into the body. And what are the body parts of the organism called as? Antigen. So sir, at least you must remember, it takes 1 week for antibodies to form. Antibodies are made later. So sir, what is the first thing I can detect in any disease? Always antigens. Antigens are the first things to be detected. So if you see less than 5 days, if I want to do the diagnosis, the best thing I can do is PCR. Best is PCR, followed by detection of one part of this virus called NS1 antigen. NS1 antigen. Next, more than 5 days if you're talking about, then I can do the test called ELISA. ELISA. So sir, first antibody as usual IgM, later IgG. ELISA will detect this antibodies. So antibody ka detection, always remember, happens at least after 1 week. That's why in typhoid also, when you do Vidal test, when is Vidal test done? After 1 week. After 1 week, because antibodies only come after 1 week. Okay, that's it. That's your dengue virus. Chaliye, now you can recognize these lesions one by one. Okay, so here we go. Unilateral lesion. Unilateral lesion, this is nothing but shingles, right? Shingle is single. Next, this is chicken pox. If you see, this is very easy. Owl's eye appearance of CMV, cytomegalovirus. Cytomegalovirus. Slap cheek appearance of parvovirus. Last and the final, you're seeing dome-shaped lesions with central depressions. That is called molluscum contagiosum. Okay, molluscum contagiosum. So that's it that you got to know right there, my friends. Okay, done. Now moving further to mycology. Sir, fungi, their study is called mycology, as you know. Fungi more or less exist in two forms, either yeast or mold. What is the speciality of yeast? Yeast produced by budding, right? This is the original yeast cell, then a small baby called bud comes out from it. That is what is budding. Next, molds are going to produce this long, structures called hyphae. Molds produce hyphae. Now, there is somebody who's trying to look like molds. It's originally a yeast, but it wants to look like molds. So, this is a yeast, but the hyphae it produces are elongated. That kind of looks like the hyphae. Okay? The buds here, they are so long, they look like hyphae, and that is done by Candida. These are not true hyphae. It's a yeast. It's a yeast producing the buds that look like hyphae. They are fake. That's why they are called pseudo hyphae. Pseudo hyphae is a feature of Candida, very important. Okay, pseudohyphae will be the feature of Candida. Done. Next, dimorphic fungi, the name says it all. Di means two forms, dimorphic fungi, two forms. They change their shape based on the temperature. That is something we learned in the classes 37 and 25 degrees. Anyway, now coming to the different types of fungal infections. First one is called dermatophytes. Okay, first one is called dermatophytes. Derma. Derma is the first keyword that we will be using here. Derma means skin. So you must remember that all the three in this will involve skin. Skin to hoga hi hoga. Dermatophytes, also called by some other names, all of them involve skin. There are three types of dermatophytes: trichophyton, microsporum, and epidermophyton. Now, trichophyton mein the hint we take, I told you during classes, is skin, hair, and nail. Three things are present: skin, hair, and nails. Three things are involved. Next, sir, if you talk microsporum, in microsporum, skin and hair are involved, and epidermophyton, skin and nail are involved. ठीक है, what is the hint I used to give you? Epiderm को imagine करना है. Epiderm means outside layer. So you got to remember that skin and nail is epiderm, outside layer. Epidermophyton is skin and nails. So first thing is done. ठीक है, knowing which tissue is involved here. Next thing is macroconidiae. These are some structures of this fungi that take some shape. One is pencil shape, one is the club shape. So what is the mnemonic for this that we generally learn? Okay, we used to learn the mnemonic called epic trip. Okay, epic trip. So what is the mnemonic epic trip teaching us? Check this out. Sir, in trichophyton, you have pencil shaped. Trichophyton में pencil shaped. Epidermophyton में club shaped. Epidermophyton is club shaped and trichophyton, they are pencil shaped. That's it. Next, moving to the other types. Another type of tinea infection is called tinea versicolor. It is caused by a fungus called Malassezia furfur. It is the guy that shows spaghetti and meatball appearance. This fungi is going to show you the spaghetti and meatball appearance. Okay? So please remember, आप टीना के साथ date पे गए हो. See, make up a story stay in your head. You have gone on a date with Tina. Tina is wearing a jacket made out of fur, and on the date you guys have ordered something fancy, spaghetti and meatball. Spaghetti and the meatball. So this is it. You can see this description right here is described as the meatball. This is the spaghetti like appearance, noodles. All right. Next, similar thing right here. This is the meatballs. The other things around, these are the spaghettis. So spaghetti and the meatball appearance. ठीक है, so Tina के साथ on the date, she's wearing a fur coat and you have ordered spaghetti and the meatball. Okay. Next, moving to infections below the skin. These were all on the skin, remember? Now we are moving to the infections below the skin called subcutaneous. One is a guy called as chromoblastomycosis. This produces warts like HPV, right? This lesions will look like HPV. Now this is the guy that is having chrome in the word. Chrome means color. We are going to remember one color, copper color. Copper penny appearance known as sclerotic bodies. Chrome matlab color. Chrome means color. Copper penny, that is sclerotic bodies. So you got to remember this. Next, sporotrichosis, also known as rose gardener's disease. Rose gardener's disease. See, rose gardeners means roses we are talking about. So you will get this disease through a thorn prick. So that means this thorn will enter inside like this, thorn prick. So what happens? You will get an ulcer in that area. But unfortunately, wherever you get pricked by the thorn, you're not going to have a lesion only there. It starts spreading here and there. Through the lymphatics, that lesion starts spreading here and there. That's what is going to happen with sporotrichosis. Lymphatic drainage is going to move around. Answer, you see the star-shaped bodies which are given a name called asteroid bodies. Star-shaped bodies by the name of asteroid bodies when it comes to this. Okay. Next, I have already explained to you about pseudohyphae. Pseudohyphae of Candida, one identification character. And another test is called germ tube test. You need to keep in mind, also known as Reynolds Broad phenomena. Germ tube test known as Reynolds Broad phenomenon. Next, sir, moving further. In HIV patients, when their CD4 count becomes less than 200, that means their immune system is falling. The CD4 count has become very, very less now. Usually it's somewhere between 500 to 1500, but now it has become less than 200. So this is where we get infections like Cryptococcus or Pneumocystis. Cryptococcus or Pneumocystis. You got to know Pneumocystis. Pneumo word is here, that means pneumonia. This is the guy that will cause pneumonia in the AIDS patients. And you have to remember Cryptococcus, Dr. Cryptococcus. See, Cryptococcus causes meningitis in AIDS. It causes meningitis in AIDS. And a very famous thing that you see is India ink stain. India ink stain. All right. This is the stain specifically for the capsules. All right. So India ink stain आपने रट लिया होगा. So hopefully this is okay with everybody. That dark color background against which the capsules are illuminated. That is what is this India ink stain. Last one or two things is Aspergillus. In the Aspergillus, there are multiple species. I'm just going to talk about one right now. Aspergillus flavus that produces this toxin called aflatoxin. You get a word afla right here. And this you got to remember is associated with hepatocellular carcinoma, liver cancer. It is present on things like maize, groundnuts, etc. Okay, so this is the fungus that is present on these crops, what I told you about. And that thing, if you're coming in contact with the toxin, it may increase the risk of liver cancer. Okay. Now, let's have a look at these images that are here. I've already explained to you spaghetti and meatball. I hope you remember this image, very classical mycetoma. Mycetoma also called Madura foot. There are two types of organisms who can cause Madura foot. One is bacteria called actinomyces. Hence that bacterial mycetoma is called actinomycotic mycetoma. Another one is fungal one. Fungal one is called eumycotic mycetoma. Now these are those copper penny bodies that you're looking at. This is the ulcer spreading along the lymphatics, that is sporotrichosis. And lastly, both of these pictures are India ink stain. That means against a relatively darker background, you see the capsule being illuminated. So ये सब capsules है what you're looking at. Okay. So this was India ink stain. Next, you have Aspergillus and mucormycosis. See, presentation of mucormycosis everybody must know. Mucormycosis, first the risk factor is diabetes patients, especially diabetes type 1 patients who are having DKA, diabetic ketoacidosis. It is a very important risk factor for this, or at least the word diabetes you should relate. Steroids obviously increases the risk of infections because steroids reduce immunity, increasing the risk of infections. Okay, so steroids abuse or this diabetic patients, they're more prone to mucor. Now this picture is showing us the mucor. Okay, it is showing us the mucor, and this picture is for the Aspergillus. We are looking at their hyphae. Both of them are molds. Mucormycosis and aspergillosis, both of them are molds related infection. Now in the mucor, this is the hyphae. It branches at roughly around 90 to 120 degrees. So this is the branching angle. Whereas if you see in aspergillus, it is 45-degree branching. 45-degree branching here. And you got to know something, that is, if you see Aspergillus carefully, there are some lines present called as septate. Septate hyphae is a feature of Aspergillus. Septate hyphae is a feature of Aspergillus. So sir, the way I teach and I ask you guys to remember this is basically, if you check this out, imagine the letter A for Aspergillus. Same A you turn around like this. Now you're getting the letter 45 degree. So this 45 degree is your fingers. Look at your fingers, you're seeing your septate. You're seeing septate. That means 45 degree with the septate. And finally, the drug of choice we memorize by the name of voriconazole. Voriconazole. So Aspergillus, 45 degree with the septate, and finally voriconazole. Exactly opposite is mucormycosis. Aseptate, not 45, it is 90 degrees, and drug of choice is amphotericin B. Anyway, that is the pharma part, but still amphotericin B or liposomal amphotericin B, whatever you call it, basically that's the drug of choice for this. Okay, mucormycosis, this is the drug of choice. Last May, you're looking at the conidae now. You're looking at the conidae. Now what you need to know is, this is the conidae of somebody called Aspergillus. Conidae of Aspergillus, this is a different structure you're looking at. And sir, this is the conidae of somebody called Penicillium. Penicillium is also now called as Talaromyces. Talaromyces. Okay, so you got to remember something here. When we are sharpening our pencil, the wastage that falls down, you have to clean it now. Pencil का जो गिरता है ना बुरा. So when you are sharpening your pencil, the waste that will fall, you need to clean it. You need some brush, you need some broom. So this one looks like that brush or the broom. So brush like conidae, झाड़ू देना होता है. Pencil का बुरा जब गिरेगा, you have to brush it off. You need to broom it off. So basically, brush like conidae is seen in Penicillium. And this Penicillium or Talaromyces produces red color pigment. Very important. It produces red color pigment on the culture media. Culture media. All right. So that's all about this, gentlemen. So we got this idea. Up the next section, parasitology. Okay. Now you all know that parasito is broadly divided into two main topics. One called as protozoa, and second one called as worms, helminths. So first we will start with protozoa. There is somebody called entamoeba. We studied this guy in the Go Get On Metro mnemonic. Remember entamoeba, that is the guy. It enters your body through something called quadranucleate cyst, which is written below here also. So there are two parasites, entamoeba and giardia, both enter your body by quadranucleate cyst. Obviously through the fecal-oral route. If they're entering through fecal-oral, mostly they may cause diarrhea. Both of them cause diarrhea. Dysentery means a bloody diarrhea. This gentleman causes bloody diarrhea. And this gentleman causes fatty diarrhea, steatorrhea, steatorrhea. Now when you look at giardia picture, I'm going to show you, this guy will have a human-like face. Human-like face. Okay. So now you got to know something. This guy is having human-like face, and it's roughly going to look like a leaf, triangular leaf kind of a structure. Okay, so you got to know this. It is having a leaf-like structure. So sir, when it moves like a leaf only. Falling leaf motility, which we talked about. Okay, falling leaf motility, it is seen by giardia. Now that we have done this, okay, let's move on. Cryptosporidium, isospora and cyclospora. Okay, Cryptosporidium, isospora and cyclospora. Now this guy, new name is called cystoisospora, but who cares? Cryptosporidium, isospora, cyclospora. From this, the most common agent is Cryptosporidium. It causes diarrhea in the AIDS patients. So entamoeba, bloody diarrhea, giardia, fatty diarrhea, and this guy is causing diarrhea in the AIDS patients. And when you take the sample, their cysts are going to stain with our special stain called acid fast stain. Acid fast stain. Are you understanding this? Acid fast, anything will appear deep pink in color. Okay, deep pink in color. So this is what you got to know. Cryptosporidium, Isospora, and the Cyclospora. Next, once again, another guy from the Go Get Back on Metronymic, Trichomonas. This Trichomonas is the guy that is going to cause the STD that presents with a greenish colored discharge. So you can see right there, it is presenting with a greenish colored discharge. So please remember, usually these guys have two stages, trophozoite and cyst. Two stages they have. But this guy does not have a cyst stage. The only stage is trophozoite. So Trichomonas में सिर्फ trophozoite होता है. It does not have cyst. This is a previous MCQ also, by the way. ठीक है? So Trichomonas have only trophozoite. It does not have anything else. बाकी greenish discharge. I have told you one more thing, that is metronidazole is the drug that we need to use and treat both partners. And sir, when you do the colposcopy, you're going to see strawberry cervix in this. Definitely in the gynae, you might have talked about this. It's going to present with strawberry cervix. Fine. Next, an organism called Leishmania donovani. Leishmania donovani. So Dr. Saab, this is the guy that causes a disease called as Kala Azar. Kala Azar. You must know that how does the Kala Azar disease comes in our body? Through the bite of a sand fly. It's not a mosquito, it is sand fly. And if you want to diagnose this disease, you got to know that there is something called LD bodies. LD bodies, easy to remember. Leishmania donovani diagnose karne, we have LD bodies. And one antigen we can detect called RK39 antigen. Keep these two things in your mind, please. RK39 antigen test and LD bodies. LD bodies. Fine. Now, the next table that I have is the table of malaria. You could have already figured just by looking at these two words, Plasmodium vivax and Plasmodium falciparum. Okay. So these are the two things, Plasmodium vivax and Plasmodium falciparum. Now, Sirji, Acol forms, also called as applique forms. These are certain ring shape of plasmodiums that is seen at the corners of the RBC. Corners of the RBC. All right. Usually I tell you that they look like headphones. So headphone forms we used to learn like this. They are absent in vivax. These headphone forms, they are present in the falciparum. They're usually seen at the corners of the RBCs. Next is the shape of gametocytes. It is banana shaped gametocyte when it comes to falciparum and simply circular in vivax. In the RBC, you get to see some dots called as Maurer dots. Maurer dots in falciparum versus this. So all this story I'm going to explain to you, obviously. Now, the most important thing what they can give you in a peripheral blood smear of malaria patient is, if you imagine this is the RBC, okay? If you imagine these are the RBCs that you are looking at. In the vivax, there is only one single ring like this, whereas in the falciparum, there are multiple rings present. Okay? In the falciparum, there are multiple rings that are present. So, Doctor, for falciparum, you can remember one mnemonic called BMAR. BMAR means sick. Falciparum is the guy that is the most serious malaria causing agent. He's the one who's going to make you sick. Falciparum makes you sick. That's why the mnemonic we are using is BMAR. What is the BMAR mnemonic? Let's learn all the points of falciparum right here before exam. First one, B for banana shape. Banana shape gametocyte. Okay. Next one, Maurer dots. I just told you, Maurer dots. Next, Acol forms present. Acol forms present. Next, this guy affects all aged RBCs. It doesn't spare anybody. It affects young RBC also, old RBC also. Vivax affects young RBC. Vivax and Ovale, they affect only young RBC, whereas this guy affects all aged RBC. And there is somebody called Plasmodium malariae that usually affects old RBCs. Okay. Now there is one more word. This guy has a chances of coming back again. It's called recrudescence. Recrudescence. So this recrudescence is positive with which one? It is positive with this guy called as falciparum. So you got to know this main things when it comes to falciparum versus this. I'm going to show you the image coming up shortly about this guy. Okay. So done. इसका treatment भी अच्छे से याद रखना, whatever we used to learn during the classes. Okay. Vivax versus falciparum along with the mnemonics I have taught you in the classrooms. So coming to the worms now. In the worms, the first worm I'm going to discuss is schistosoma. Now, schistosoma, you got to remember it causes bladder cancer. And which is this bladder cancer? Squamous. Schistosoma causes squamous bladder cancer. Clonorchis causes cholangiocarcinoma. ठीक है, S for schistosoma hematobium, S for squamous bladder cancer, C for clonorchis, C for cholangiocarcinoma. Cholangiocarcinoma. ठीक है, so that's it that you got to know. Next, sir, Taenia solium, Taenia saginata. These are some other worms. Taenia solium is called as the pork tapeworm. It's called as pork tapeworm. You got to know that this is the worm that is found in the pigs, hence the name pork tapeworm. So technically, you can get this worm inside your body by eating pork, which may usually just give you a diarrhea and you're okay. But sometimes ingesting the eggs of this parasite, okay, ingesting the eggs, it need not be in the meat or anything, it may be in anything, contaminated food and water, we may get the more worse form of the disease called neurocysticercosis. As the name says, it's going to form cysts in your brain, and doctors, this is going to manifest as seizures. Okay, so please remember this. If they are giving you a history, case scenario of a seizure, etc., you got to know Taenia solium, neurocysticercosis. And in pharma, you got to know that a drug called albendazole is the drug that we prefer here for the treatment. Albendazole along with the steroids. That's what we use. Okay. Next thing, Echinococcus granulosus. It is dog tapeworm. Dog tapeworm. So you got to remember that this was the guy for which I told you something called Cassoni's test. Cassoni's test. Remember, it's an example of type 1 hypersensitivity. So these three main things you got to remember together. C, D, E. Cassoni's, dog, Echinococcus. This is the guy that will be causing a disease called hydatid cyst disease. Hydatid cyst disease. You're going to see certain cysts forming in the liver. That's it. That's your hydatid cyst disease. Next worm is Enterobius vermicularis. This is the worm that is going to cause perianal itching. Okay, this is the worm that is going to cause perianal itching. Now imagine the anal area. You have a great division in between, yes or no? So same like this, the egg of this parasite looks like this. It looks like a coffee bean, to be more precise. You can simply remember, like if you're looking at the buttocks, there is a line in between. It's causing the itching in that area, right? So the egg of this guy looks like that. Coffee bean-like thing. Next, hookworms. There are guys called Necator and Ancylostoma. These are the ones that will be causing iron deficiency anemia. Hookworms, they are the blood-sucking worms basically. They will be causing iron deficiency anemia. Fine. Next is the filarial worms, Wuchereria and Brugia. There are two, Brugia malai and Wuchereria bancrofti. These are the guys that are spread by the mosquito called Culex. I told you this before. Japanese encephalitis and filariasis is spread by Culex mosquito. And what you got to know here is this. When you collect the blood smear exam, you will be examining the tail of these worms. If you see 2 to 3 nuclei in the tail, then that is Brugia malai. Malai is something we get on the top of the milk. Yes or no? That fat is nothing but malai. Malai is an extra thing. So this guy has extra things called nuclei. Malai is the extra. Next, Wuchereria bancrofti, bankrupt. Bankrupt means it does not have any nuclei. Okay, so Wuchereria bancrofti, no nuclei, whereas this guy has nuclei in this. Usually these worms do not come out during the daytime, they come out only during the night time. Okay, that's why there is a word nocturnal periodicity. So if you want to do the testing, take the blood sample during daytime and expect the worms to be there, you have to provoke them by a test called DEC test. Okay, there is a drug called diethyl carbamazine that we give, which will provoke these worms to come out. That is what we are doing right now. And that is what is called DEC provocative test. Fine. So now that's it that you had to know when it comes to this. Up, sir, let me just show you some parts in this. Why don't you guys solve some questions over here? See, a 32-year-old female has a complaint of discomfort while peeing. She also described her genitalia feel sore and red. She usually feels pain after intercourse and passes a discharge. A sample was collected and the picture stain shows this. What is the infection that you can expect? Sir, pretty much every option I have given there, all of them present with discharge only. All the 4 options present with discharge. But you're looking right now at a trophozoite. You're looking at a trophozoite or somebody. Trophozoite of Trichomonas. Same purple but having human-like face. Okay, human-like face. That is going to be Giardia. Giardia is going to be like that. Okay, that guy is leaf-like, having falling leaf motility. But why would that guy come here? Because we are talking STD, genitalia problem, hai na? So that's why this is the answer over here. This is the trophozoite. Okay. Next, moving further. This is your second question. A person, 17-year-old male, is brought to the hospital due to fever, frontal headache, photophobia, nausea, and anorexia. Physical exam shows positive Brudinski sign, meaning it's a confirmation of meningitis. Sir, LP was done showing so and so. Gram stain of the following organism is given below. What is the organism? Sir, you got to remember this. Pink color organisms round are seen. We did gram stain today only. What did I tell you? If you see pink and round, then that's Neisseria. Neisseria. Another hint that was given. Usually up till 17-18 years, we have Neisseria. 2 years to 18 years. In this window, Neisseria is the guy that causes meningitis. Okay. So that another hint that they have given in this question. So sir, at least as a quick recall for the exam, listen carefully what I'm saying. Strep pneumoniae is pneumococcus. Pneumococcus is positive. This positive guy is purple. Whereas if you talk about Neisseria, Neisseria is negative and this guy is pink. Are you understanding this? Neisseria is going to be negative, it's going to be pink. Pneumococcus is positive, it's going to be purple. Both are cocci only. Both are cocci. So color can differentiate them. Positive means purple and negative means pink. What I told you in the gram stain. Okay, that's done. Now, Doc Saab, the next part what I want you to do is please remember quickly. One of the first things you should always look at is glucose, what is given in this. Not every time they will give you gram stain. If you see the glucose levels, normal is this much, this is less than that. The moment you see glucose is less, you should figure out whether it is TB or whether it is other bacteria. TB is also bacteria, but something different is there. So anyway, glucose is less means it is bacteria type of meningitis. TB is also a bacteria, by the way. right? If the glucose is normal, then it is virus. Virus mein glucose is normal, whereas here in this glucose is less. As a matter of fact, drastic changes in the normal values, if you see, it is called bacterial meningitis or pyogenic meningitis. Everything is very extremely changed, then that is this one. Now what do you need to do is look at the cells that they are mentioning here. Okay, if they are mentioning certain cells like neutrophils, then you should know it is bacterial one. If they are mentioning lymphocytes, then that is TB. तो ऐसे differentiate करना है. First you look at glucose. Glucose is less TB or bacteria. How do you figure out that? Lymphocyte or neutrophil. In this case, unfortunately, they didn't mention that. That's why I have given you gram stain. My aim was to make you recognize this by the gram stain. Okay, so correct answer will be Neisseria for this. Okay, I hope everybody is clear with that. Fine. So this is the way you're going to differentiate this, my friends. Fine. Next question right here. A 35-year-old female presented to the hospital with complaints of loss of appetite, nausea, joint pain, fatigue. Physical exam reveals yellow sclera and dark urine. She doesn't drink alcohol. Serology report shows something positive, something negative. What is the interpretation of this? Okay, see, obviously something what we have studied. The initial parts of the question are just describing she is having obviously hepatitis. Those are all the presentation of hepatitis. If you're having inflammation of liver, you will be having loss of appetite, nausea, vomiting, this kind of symptoms will be there. They confirmed it by giving yellowish sclera. They said she doesn't drink alcohol, so most likely it cannot be alcoholic hepatitis. It is viral one, hepatitis B. So अब बता रहे हैं, they did a test, something came positive. As long as HBs antigen came positive, the person is having disease. If you see anti-HBC is IgG, then it is chronic. It is chronic. And third one, she is infectious also. So chronic infectious hepatitis with high infectivity. This is the correct answer for this. Chronic infectious hepatitis. Okay, done. Next one, Sergi. A 40-year-old male with history of rheumatic heart disease is admitted with fever, cough, shortness of breath, splinter hemorrhages, and clubbing. An echocardiogram shows evidence of vegetations on the valve. Okay. He recently underwent a dental procedure. The organism was found to be catalase negative and alpha hemolytic. Which of the following organisms is most likely to be grown in the blood culture? Okay, which of the following is most likely to be grown in the blood culture? Okay, so you need to pay attention. What are they hinting you? First of all, this disease is endocarditis, infective endocarditis. 90% there is a chance there will be a question on infective endocarditis in your exam, something or the other. Okay, so question said catalase negative. The moment they said catalase negative, option A and B cannot be correct because it is catalase positive. Remember, staph is going to be catalase positive. Next one, moving on, they mentioned alpha hemolytic. I've already told you. अब बचा ही एक ना sir. The only guy that is left catalase negative is Streptococcus. Streptococcus viridans. Streptococcus viridans. This is the thing that you need to remember here. Okay. So technically the answer to this boils down to Streptococcus viridans. Please remember, Streptococcus viridans are the most common organisms for subacute bacterial endocarditis. Okay. Subacute bacterial endocarditis. This is the first thing. And second, you got to remember, they are even the organisms for late onset prosthetic valve endocarditis. ठीक है. So for these two, the most common organism is Streptococcus viridans group, like sanguis. There is somebody called Streptococcus sanguis that comes under this. Okay, so you got to remember ये answer यहाँ पर होगा. If they're asking early onset prosthetic, early E for E, epidermidis. Okay, this is the guy for early onset. Early onset का it is epidermidis. Fine. If these words are mentioned, then you will be choosing this answer. Nothing mentioned, one answer. The overall most common organism, one single guy you have to answer for endocarditis, whether it's an IV drug abuser or not IV drug abuser, I don't care. The most common organism is Staph aureus. Okay, the most common organism generally found in the cases of endocarditis, it is overall Staph aureus, the superstar. Up they are mentioning specific words and they're expecting you to answer some other things by giving tests like this, then you have to choose the answer accordingly. Okay, then that's how it's going to go. You have to choose this accordingly. Okay, friends. So this is it. This is your endocarditis wali story. Now, anyway, since we are done with this, now guys, please follow me on Instagram and Telegram, especially now the people who are not there, because you know I have shared one schedule where I have asked you to revise one small topic each day, you know, usually from micro as well as pharma, and I'm posting questions once a week on those topics. So that way at least you get to revise these things. All right. And once you join me on my Instagram, you will be seeing more links for the YouTube videos where you get, you know, more questions, etc., with their explanations that are expected in exams. So definitely join me. You're yourself going to be benefited out of this. Okay. Baki, that's it from me. Any queries that you have, guys, this is just a rapid revision session. So any queries that you have, please feel free to message me on the Instagram. I will respond back to all of you. Okay, so people who are already in my list, you already know that I answer your queries. So please वहाँ पर देख लेना. Whatever it is, this was planned to be done in this session so far. So this would be from me. Please shower your love in the comment section. Share this video with your friends also, so that they also get to benefit something out of this. Okay, these are very highly anticipated things that we have done. Okay, so that's it. Expecting, you know, your comments that you got to learn something from this. Thank you very much. Good night. Shab Khair. Namaskar.